Here to welcome you all in the first technical session of the National Seminar on Fungal Systematics and the Microbial Exploration. This seminar is jointly organized by the Science College Nagpur, Mahatma Phule Arts, Commerce and Sitaramji Chaudhary Science College Varun, and the Mycological Society of India, Amravati chapter. And we are delighted to see such an enthusiastic response from the scientific community. Today's first technical session focuses on one of the core areas of the mycology, that is the fungal taxonomy, and which forms the foundation for the understanding biodiversity and the ecology and the applied type of research. It is our privilege to have with us Dr. Amit Pal Singh ji from the Department of Botany, Punjabi University, Patiala, who is a renowned expert in the taxonomy of wood-rotting, corticoid, and diporoid fungi. Dr. Singh will deliver a lecture entitled Polypore Taxonomy Morphological Approach. I now request our esteemed chairperson, Dr. Nitin Dongarwar, to formally introduce the speaker and conduct the session. Thank you.
Thank you very much, Dr. Ganesh. Good morning, one and all. It is my proud privilege to introduce the keynote speaker, Dr. Amit Pal Singh. Dr. Amit Pal Singh, he has passed his matriculation from Government High School, Barnala, BSc from SD College, Barnala. Dr. Singh has completed MSc and PhD from Department of Botany, Punjabi University, Patiala, and he was awarded with the University Medal for standing first in the order of at the MSc Botany level. He joined Department of Botany, Punjabi University as a faculty member in the year 2011, and he has been a member of undergraduate and postgraduate board of studies in botany in various universities and colleges. Dr. Singh is working in the field of systematics and taxonomy of wood rotting and corticoid and poroid fungi. He has supervised 8 PhD students for their doctoral treatise, 2 students for MPhil, and 23 students for their MSc degree. He has successfully completed a major research project funded by SERB, Department of Science and Technology, Government of India. He has published more than 80 research papers in the journals of national and international repute. He has described more than 250 tags of corticoid and poroid fungi from Punjab, Himachal Pradesh, and Jammu and Kashmir region of our country. He has to his credit 3 new genera, 23 new species of corticoid and poroid fungi. He is a life member of Microbial Society of India, Punjab Academy of Sciences, Patiala. He has also served as a counselor for the year 2014-2017 and managing editor of the journal Kavak. He has attended more than 40 national and international conferences. He has organized national conference on biodiversity and biotechnology of fungi and 47th annual meeting of Mycological Society of India, and has been awarded with Professor K.G. Mukherjee Memorial Award and Professor P.B.R. Vittal Memorial Award for Middle Age Scientist by the Mycological Society of India. So I proudly present Dr. Avneet Pal Singh, and I request him to deliver his much awaited talk on polypores.
Thank you so much. Thank you. So I'm extremely grateful to Dr. Dongarwar and Dr. Hidavu for those wonderful words. And they have been so kind and always have that positive energy to infuse with their polite words. Let me start my talk by paying tribute to that great visionary, Dr. Punjab Rao Ji Deshmukh. Punjab has got that beautiful connection with this great visionary. There's a name Punjab in his initials, and we always remember him for his contribution to the field of agriculture and for organizing that beautiful sector of agriculture across the country. And I also bow my head in front of Rashtra Sant Tukadoji Maharaj and Sant Baba Gadge, the kind of contribution to the field of education that has been made is immense. So I'm thankful to both the conveners, the organizing secretaries, the co-conveners and the chair for giving me this opportunity to interact with friends from Vidarbha. And let me start my talk by sharing a recent experience. Me and Dr. Hande, we were recently in the forest of Pench Tiger Reserve, and while we were wandering in that forest, we could still locate a good number of polyporoid mushrooms growing on those beautiful wooden logs, the mature trees, and the fallen wood. And we collected a couple of them, and that promised that yes, this association can grow further. And the kind of diversity that has been there, mind it is the off season for fungus. There is least moisture present in the forest. The temperature is dropping down. It's below 20 degrees Celsius, but still those beautiful creatures are growing and they are displaying great diversity in their morphology. So that prompted me to name this talk as the morphological approach as far as the taxonomy of these polypores is concerned. Can I have the next slide? Now the name polypore, poly means many. In science we have that habit of splitting the words and explaining them to our students. Pores are very fine openings. So in the forest, dear friends, you see some fungal fructifications, the macroscopic fructifications which have some sort of openings present on them. And because of that, these fructifications are called as spores. They are many in number, so we call them as polyporoid fungi. So they display a lot of variation, forming very fine sheets to somewhat raised on their margins and finally assuming that three-dimensional beautiful pileate forms. Ganodermas, we all are aware of. Felinus, we all know. Inonotus, these three names, they come to our mind when we think of this group of fungi. In the forest ecosystem, their major contribution is recycling of the logged carbon. They can decay cellulose, hemicellulose, and yes, lignin. And they cause those two beautiful type of rods, the white rods and the brown rod, releasing that logged carbon back into the ecosystem. And on the same time, some of them, they are exceptionally medicinally important. And when you search the traditional knowledge systems literature, you find n number of applications of these fructifications in powdered form, extract form, and they have been used since ages in the traditional medicinal system. Now, if you want to identify them, let's first briefly understand how do they develop. There is one major difference between the lamellate mushrooms, which are of course edible, polypores, few of them are edible, majority of them they are non-edible. They form very thick leathery fructifications which are difficult to chew. So if you see the development of a polyporoid basidiocarp, you will always see some sort of initial development in the form of some mat on the woody substrate, which is otherwise absent in lamellate mushrooms. They suddenly appear, they suddenly disappear. In this case, you will see a visual fructification, pre-fructification phase on the woody substrate, and gradually it will develop and will produce that beautiful fructification which varies in their form, sheet-like or typical pileate. Next slide, please. Now, whenever we see a poroid fructification, we have to think about two types of morphological characters. Some set of characters, they are visible in field. You don't need any aid to observe them. It is just your keen observation and your eye, sharp eye, which is required. And dear friends, you can reach to some kind of level as far as their identification is concerned. In some cases, even you can reach up to genus level by just carefully looking at the fructification. So first, let's have that discussion on the kind of macroscopic morphological characters, the macromorphological features. Now, first thing, we should note down what kind of basidiocarp is being produced. Is it sheet-like? Is it reflex? Is it pileate? Then we should see what are the different kinds of developmental patterns. When you will see a fruit body, if there is a single layer of pores, that is a typical annual fructification. And when you see carefully in certain species of Ganodermas, Felinus, you will see layer after layer developing, and those are developing every year. So they form typical perennial fructifications. There is a lot of diversity in their shape, in their nature, and in their annual or perennial forms. This is the first thing. If you have to apply a taxonomic key, that should come to their mind. Second is mode of attachment. Broadly speaking, we have two modes of attachment. Fructification directly attached to the wooden substrate, there is no stipe, and in botany or basic sciences, we call them as sessile fructifications. Or with a very defined stalk, we call them as stipitate. And sometimes there is an intermediate stage we often refer to as subsessile or substipitate kind of situation. And in some cases, what happens? Base of the fructification, pileus, that gradually narrows down. And by that narrowing base, it becomes attached. If you note down these four features, the first phase of your morphological observations that has been carefully taken care of. Next slide, please. Now then, there comes a little bit of technical part. Now here, there is a beautiful publication, Glossary of Fungi. It was published in 1979. In that beautiful Glossary of Fungi, some technical terms have been explained. So, from those technical terms, people have taken those features in their taxonomic keys. So I can share the soft copy that is now out of copyright with the organizing team, and you can have a look on that Glossary of Fungi. From that Glossary of Fungi, what we take into account is the shape and nature of the fructification. If you see the picture A on the left corner, there is a log that has been shown, and on that log a sheet has been produced. We call it as resupinate fructification. B and C, they give you the kind of appearance of stipe. Then there is from D to M, there are plethora of shapes. Is it triangular? Is it spoon-like, spatulate? So second thing, we have to look into the fructification shape. That is very, very important. And in case of polypores, shape is a very, very stable character. Sometimes the species of a genus, they are differentiated from one another on the basis of shape. In Ganoderma, for example, we have one species, Ganoderma lucidum. There is a typical reniform shape of the pileus. It looks like the outline of a kidney-like structure when you look at Ganoderma lucidum. And on the other hand, Ganoderma colossus is typically spoon-shaped. So from that very careful observation, right in the field, you can make out your rough notes that I have to compare this collection with Ganoderma lucidum or with Ganoderma colossus. That is the importance of the shape. Next, please. Then you have to look at, this is all I have explained, perennial and annual. And this is, see, in that picture H, there has been 1, 2, 3, 4, 5, 6 layers of pore tubes they have been produced. So what we do, we simply cut the fructification pileus from top to bottom with a simple blade or a cutter if it is little bit hardy, and you will come to see number of layers of the pores that will confirm your observation whether it is annual or perennial. Next, please. Then you look, these are the various common shapes which we generally use in the terminology of polypore identification: pendant, spatulate, ungulate, trichoterous. They all have been beautifully explained in that Glossary of Fungi. Then there is consistency. I have earlier mentioned that these fructifications are not edible, they are not soft, their consistency is hard. They are leathery in nature. So second thing we have to note down is the consistency, which is generally woody in these cases, with some exceptions. Now consistency depends upon hyphal construction. As students of botany, we all have been going through those basics of mycology. What is the number of type of hyphae present that will define what we call as hyphal system? If there is single type of hyphae, monomitic. Two types, dimitic. Three types, trimitic. So if your fructification is very hard, very tough, woody in nature, it is indicating that it is consisting of minimum three types of hyphae. So from consistency, you can make out what will be the type of hyphae you will be looking for when you will prepare your micro slides. So this is an important feature which forms that foundation for further explorations. Next one. These are all shapes. Next one, please. Now see, this one picture gives you the entire range of shape and nature. In the left hand bottom, you can see there is a sheet and sheet is gradually reflexing from the margins. And in other cases, you can see typical three-dimensional fructifications, they are coming out. These are the two major forms which you will be generally observing as far as the shape and nature of residiocarp is concerned. Next, please. See, then you have to see whether your fructification is produced singly or it is in clusters. This is another feature. For example, in that top right hand side fructification, you see one huge fructification. This is the typical consistency of some species of genus Inonotus. You will see huge fructifications with somewhat brownish colored lower surface. The bottom one is Ganoderma lobatum. It typically produces broadly attached single fructifications. This is Inonotus mikadoi, which produces very small fructifications, and wherever they are produced, they will be produced in huge numbers. So from the field, you are getting an idea which genus to move in, which particular family to look in, and which particular character to be concentrating upon. Next, please. See, in this case, these are typically imbricate clusters. The fructifications are coming one above the other, right like that. And this is again a character which we see in some species of Fomes and some species of genus Felinus. Next, please. Then the feature pertaining to any sort of fragrance and taste. Majority of polypores, they are tasteless. They do not have any defined taste on them. They do not have any defined fragrance except for this species of Trametes suaveolens. Wherever there is a term suaveolens associated, there is a sweet anise fragrance that is there. So this is the only thing which we see. Generally, they do not have distinct taste or distinct smell. There is another species, Hapalopilus nidulans, the bottom one, that has the only edibility. The rest of the polypores, they are inedible. And the middle picture is Polyporus latiforus sulphureus, the chicken of woods. This is the species which has got that consistency and it is highly nutritious. Another important one, polypore. Next one, please. So these are the first phase of characters which we have to look upon. Then there comes the basidiocarp construction. You have collected these specimens, you have brought to your laboratory, now starts the second phase. Now, as far as the field characters are concerned, it is not easy to note down all characters in the field. Then the question comes in mind, how to preserve those characters of fresh specimens. I'll always carry a good camera. Second thing, whenever you have to click the photographs, it should be without flash. Third character, important, you should always carry a metallic scale with you. You have to put on a scale bar whenever you have to click a picture. And these pictures clicked in the field, dear friends, they help you a lot when you are solving your identification by following some taxonomy keys. And these good photographs, they even help you to compare the color change. When you bring it to the lab, gradually they will be drying upon, and if there is any change happening after that, that is also comparable. So you have to click photographs of fresh material, and then you have to click photographs of dried materials. That helps you to make very good plates when you have to either publish or submit your dissertation work. Then comes the basidio-carp construction. Now you need a good hand lens, and you have to sit on your desk and have a lot of patience. As highlighted in the inaugural session, there is an issue with loss of patience in our generation. We are eager to have the results as quick as possible, and microscope demands a lot of time to sit and to observe. More you sit, more you observe, more you learn. That is the basic fundamental of taxonomy. And more number of specimens you examine, there will be greater interest involved in the taxonomic studies. So that is the kind of passion you need. Suppose you have collected a specimen, it is having a pileus. First thing you have to look upon the upper surface. Upper surface, which is usually colored, is called as pileus surface. Lower surface, where you have fine openings, is called as poroid surface. And pileus surface, that is very distinct and stable character in case of polypores. Generally, in lamellate mushrooms, there is very quick decay because they consist of only one type of hyphae. Majority of polypores, they consist of more than two types of hyphae. Their shape is very stable and their color character is also very stable. Now, many species, the pileus has distinct color and it remains unchanged. You have to observe that with a hand lens. There is presence of very complex polyphenols, and those polyphenols, they are quite resistant to any sort of natural degradation. That is why their color remains stable. In some cases, there will be changes. We have to note those changes also, how the things happen when we try the fructification. General rule is, if you collect white and light colored fructifications, general tendency is they will become darker upon age. Why so? Because upper layer of hyphae that gradually decays and their contents are exposed and they become darker in color. In other cases, there will be no change. And when you see perennial basidiocarps, in those perennial basidiocarps, there will be zones of light and dark colors that will be very often visible when you see those fructifications. Next, please. Now the question arises, how to mention the color? In science, nothing can be arbitrary. You have to have a standard. Whenever we plan an experiment, we have that triplicate and we have to go by some standards. So for color, this is one of the best followed reference across the globe. This is called as Matthew's Handbook of Colors. It has beautiful depiction of 96 pages which are showing 12 to 14 colors variation. So you have to follow this. That is one particular range of pink and light brown color, how it varies. So you have to move across the line, you have to move across the column and just fit in your color with this particular Matthew's Handbook of Colors. Next please. Now see how color matters. I have written two words over here. These are two very important families of polypores. One is polyporaceae, other is hymenocracy. If in the field, the color of your fructification is brown or golden brown, and you have put up a drop of KOH solution, potassium hydroxide solution, and that, see, there's a small black patch which has been produced. You need not to go anywhere, just go directly to the family Hymenocracy. So you have excluded all other polyporoid families. Only focus has been on family Hymenocracy, single character color. And if your fructification is zoned, see, there are different light and dark colored zones on the upper surface. Lower surface is typically creamish white or white. It is an indicator that this specimen will probably go to the family Polyporaceae. So if you have this much of indication, write In your field, you can segregate this. I have to look for polyperacy. This I have to look for family Hymenocracy. Next, please. See, then even color plays important role in deciding or lowering your choice to the genus also. If there is again this brown color and cracks, left hand top photograph, cracks are developing in a longitudinal fashion, a typical character of genus Phalinus. If you have this blood red or pinkish red color, genus is Pycnoporus. There is no other genus which has this type of color variation. And if you have that brown color, the typical violet brown color, that typically shows a kind of Ganoderma character. So three genera, Phalinus, Pycnoporus and Ganoderma, they are differentiated only on the basis of color. An idea that what are the characters you have to look upon and how you have to conduct your microscopic studies. Next, please. So see, this is where the color plays important role. You have noted down the color. Now you have to see that the variation on surface, how the pileal surface varies. Is it smooth or sometimes there are very fine hair present on it? Is it concentrically zonate or it is azonate? These are the other features you have to look at. For example, in Ganoderma, there will always be formation of a very fine cuticle. A layer will be produced on the pileal surface. And if you examine that pileal surface layer, you will get some idea about the type of group where to venture in for further identification. So this nature that is playing very, very importantly. Presence or absence of hair, presence or absence of concentric zones, and other characters, presence or absence of cuticle. These are the features which you can see very well with the help of a lens. Next, please. See, in this bottom, you can see there are some fine hairs which are shown on the picture P. This is a typical hirsute kind of. In English, the term hirsute indicates some type of hair they are being present. Next one, please. Now see, these are the two fructifications. They belong to genus Piloporia. In one fructification's left hand top photograph, you see the presence of very fine hair, radially arranged. This is Piloporia pectinata. In other, there is a glabrous surface, no hair are present. This is Piloporia pectinatus. So this kind of distinction that helps you to get an idea which particular species you have to look upon. Next, please. See, these 6 pictures again can be distinguished only on the basis of nature of pileal surface. Let's start from left hand bottom. You see very fine hair present and it is Tramides hirsuta. You see the middle one, there are no hair present. The fructification is faintly zonate, Tramides suaveolens. You see the topmost left hand picture, alternate grey and brown zones are produced, Tramides versicolor, the turkey tail polyp. Just simple looking and giving you an idea where to look upon for your identification. On the right column, there are 3 species of Ganoderma. Now, what is the difference we are observing? Bottom one is shiny. We call it as laccate. Stipe is branched. It is typical character of Ganoderma casuarinicum. Upper surface is dull. We call it as non-laccate. Fructification is lobed. Ganoderma lobata. Middle one, the stipe is present on the lateral side. It is Ganoderma carnosum. So, just by looking on these characters, there is an indication of probable species level identification in these cases. Next one. See, a single genus Ganoderma. There are 9 species depicted in this picture. We have all these 9 species collection available with us. These are our pictures. And see how the surface varies, how the color varies. And dear friends, this color that remains stable for a long time even in the laboratory under dried conditions. So if you have an idea about the surface details to be looked upon, you can definitely end up with correct identification of your specimen. Next please. Then there comes the lower surface. Lower surface, that is called as pore surface. Because of the presence of very fine openings, we call them as pores. And two things they are to be very carefully looked upon. First, color of the pore surface, fresh and dried. Second, pore size. Now there comes a question, how to determine the size of pore. Pores are so minute, we cannot measure their diameter. Then the technique has been, you have to count the number of pores per millimeter. How much pores are there? There are two ways. Either you can have a 1 millimeter square area which is a tough task, or simply from center of the fructification to the margin, you can place your scale and under a binocular or with a hand lens, you can count the number of pores at 5 different positions from center towards the margin. Margin will always have younger pores which will be smaller in size. Center will have the largest pores which are fully developed, and the other portions, peripheral portions, can further have the decrease in size. And from that, you have to calculate the average, that will be your pore size. Third thing, shape of the pore. That is again very, very important. And technically speaking, we have lot of variation as far as the pore shape is concerned. They may be polygonal, they may be circular, they may be elongated, they may be lamellate. These are the 4 major variations which you see. Now let me explain it by taking another example. Next slide please. See, there have been 2 fructifications displayed in this slide. And this was the collection which me and Dr. Handevi saw in bench forest that was there growing still in the same position. What you can locate on the left picture bottom, pores are typically hexagonal. They have typical 6-sided appearance. In the second picture, they are lamellae-like. They look like, they are not lamellae, they look like lamellae. If the pores are hexagonal, fructifications are very thin, the genus is Hexagonia. Just look upon only for species of genus Hexagon. And if the pores are like this, just go to genus Dedilia. There are 7-8 species of Dedilia reported from India. So this has indicated which particular genus you have to look upon. So very simple observations give you reliable indication of the tentative genus and the tentative family to work upon. Next please. Then you have to look upon the 2 other characters, margin. Is there any change in color at margin on the upper surface, lower surface? Second, is the margin shape or is it sharp or it is broad? So these are the 2 things that we have to look upon. And of course, then comes the position of this type. There are 3 possible positions: central, lateral, or dorsolateral. The 3 positions are generally present as far as the attachment of ileus with this type is concerned. So if you finish with this particular set, your second phase of morphological observations, that is over. Next please. See, in this case, there is no defined margin. We can say margin is indeterminate. In other cases, there are fine fibers which are produced on the margin. We call them as margins or fibrillose. Next please. Now in this case, you see entire fructification is brownish in color, lower picture. Margin is whitish in color. So these are the minute observations that we have to look upon. When you have a pileate fructification, you have to look upon upper surface margin as well as lower surface margin. And last character, is there any pore present on the margin or it is without a pore? If pore is present, we say margin is fertile. If pores are absent, we say margins are sterile. So this is what you have to look upon as far as the margin character is concerned. Next one, please. Then there comes the second portion where we again cut the cross section of the fructification, pileate fructification. Next slide, please. When you cut the pileate fructification, you will see an upper surface which will be sort of spongy in nature. This upper portion we call it as context, and lower portion where pores are there we call it as tube layers. So previous slide, please. So there are two things we have to look upon: the character of tube and character of context. Let me first come to context. Context is the upper sterile portion of the fructification, and there are two variations. Is this layer single layered or it is divided into two zones? If it is single layered, we call it as homogeneous. If it is having two layers, we call it as duplex. These are the only variations in context region of the fructification. Lower portion is fertile, we call them as tube layers. And if you remember, if there is annual fructification, there will be one tube layer. And if there are perennial fructifications, there will be 1, 2, 3, 4, depending upon for how many years the fungus has grown. So you have to note down the number of tube layers. So this is, we can again observe with the help of a hand lens. Next, please. So same has been explained in this. Next, please. So it is a duplex. Now, only from duplex context we can get further an idea. There is a lot of confusion in identification of genus Ganoderma. Every second or third specimen from Indian collections was put in species Lucidum. And when you examine those herbarium specimens, they do not actually belong to Lucidum. And later on, it has been proved that Lucidum is a subtropical and more precisely temperate species, which does not fall in Indian context. And what we have, the other species of Ganoderma. Later on, in early 90s, there was a very exhaustive monograph on Ganoderma, and he has differentiated it on the basis of context character. So that is very, very important to look upon when we are working on genus Ganoderma. Next, please. So this completes the phase of macromorphological characters. Now you have to prepare the microscopic preparations for studying micromorphological characters. The question arises how to prepare the microscopic slides. You have taken a fructification. On lower surface there are pores. Take a simple shaving blade, topaz, etc., etc. Need not to spend on sophisticated scalpels or blades. It should be sharp, fresh blade. Move the blade parallel to the pore surface in one part of fructification. You will get a reticulum type of thing. Put a drop of KOH on the slide, 3% or 5%. It revives an alkaline medium. Put that fine cross section in that KOH. Leave it for 5 minutes. Let it revive. And after that, with the help of 2 needles, tease it. Tease it till you are teased. So that should be the level of teasing. Final is the teasing. You will see all structures in one preparation. This is my word if that teasing is finest. And then you have to put some stains. I'll come to that later. Put a cover slip. Observe under simple microscope. 10x followed by 40x and finally followed by 100x. All observations, measurements, they have to be under 100x oil immersion. That is the standard which has been used by all taxonomists in their taxonomic keys for size and other references. So when you make those microscopic preparations, you will see the beautiful world of microfungal microstructures, the hyphae. I have mentioned there can be 3 types of hyphae depending upon monomitic, dimitic and trimetic hyphal system. The group belongs to Basidiomycota. And we all know in Basidiomycota there is formation of basidia on which exogenously basidiospores are produced. So we will see hyphae, we will see basidia, we will see basidiospores. Plus, in some cases there will be additional structures. We call them as ancillary structures or cystidial structures that will also be visible, and you have to have that basic set of microscopic characters in your mind. Then you have to sit and observe the microscope very carefully. First thing, previous one please. So we have to have hyphae corner. When we study mycology literature, this person E.J.H. Corner, he introduced the concept of hyphal system. There are three types of hyphae. Generative hyphae. First formed hyphae, which develop from the germination of basidiospore, they are called as generative hyphae. All other structures of the fungal fructification, they are produced from generative hyphae. Second, next slide please. Then you have gliocorpulous hyphae. These are the hyphae which have some content, oil content. In fungi, sometimes you see oil droplets present in their hyphae. And then you have hyphae which are very thick walled, very narrow lumen, no septa are present, and they are unbranched. We call those hyphae as skeletal hyphae. They appear like solid rods. Third type of hyphae, they are very thick walled, lumen is very narrow, but they are irregularly branched, and they are again aseptate. They are called as binding hyphae. If you have a phase contrast microscope available in your lab, that is the best microscopic technique to distinguish between generative, skeletal, and binding hyphae. You have to prepare your slide in water. This phase contrast does not apply with the stained material. Either the preparation will be in KOH or it will be in water only. And in that phase contrast, you can very clearly distinguish wall thickness and lumen size. So if there is only generative hyphae present in your specimen, it is monomitic hyphal system. If along with generative, there are either skeletals or binding, dimitic hyphal system. And if all three are present, trimitic hyphal system. And dear friends, in polypores, almost all keys, they use hyphal system as the taxonomic character. You have to have this observed carefully in your specimen, otherwise it will be a dead end for your identification purpose. Next please. See, this is the diagrammatic representation. There is a beautiful monograph developed by Riverden, Leaf Riverden. He is the most authoritative person on polypore taxonomy, and he has published 7-8 monographs. The most beautiful is Poroid Fungi of Europe, where all these basics have been outlined by him. So you can look upon the variation in hyphae, the hyphae which are skeletals, bindings, generative, and you have to look upon, is there any kind of encrustation present on your hyphae. Since it is Basidiomycota, and in your basic paper of mycology, you all must have heard of that in Basidiomycota there is crozier hook or clamp connection produced, which is required for dikaryoticization in certain species. So your generative hyphae, they can be clamped also. We have to look for presence or absence of clamp connection. If dikaryoticization has already been there, there will be no clamp connection. If it happens afterward, there will be clamp produced. You have to look upon that. Next please. So this is what you have to look upon. Next please. Next please. Now then there comes the role of ancillary structures. Hyphae have been observed. Now you see certain structures which do not bear sterigmata. They are not spore-like, but they are regularly occurring in your microscopic preparations. These additional structures are called as cystidial elements. Their role is to support the hyphal structure. They give a sort of stiffness to the pileus. They give some space gap between basidia so that spores can be discharged. As basic students of botany, we always learn that in Basidiomycota there is a forcible discharge of the spores, sexual spores. So Basidia needs space for that spore discharge, and that space is occupied by these cystidial structures. So we have to look upon any of this kind of structure, but appearing more than once in your microscopic preparation. Sometimes it may be illusion, but if it is repeating 2-3 times, definitely it is an cystidial structure associated with your specimen. Next, please. See, this is the camera lucida drawing. On the left, you see a photomicrograph. We can have even this kind of very thick walled, deeply encrusted cystidial elements, and we call them as lamprocystidia. Next, please. Next, please. See, cystidia with oily contents. We call them as gliocystidia. Thin-walled cystidia without encrustation, we refer to them as leptocystidia. My presentation is here only. I have taken it from the basic. You can refer it. There is no issue, no copyright involved in that. Next one. See, the cystidia with the teeth in the center, we call it as stephanocyst. The cystidia with a bunch of crystals on its tip, we call it as asterocystidia. There is one genus, Asterostroma, identified only on the basis of occurrence of asterocystidia. See, Leginocystidia, bunch of loop of crystals on the tip, and regularly placed crystals on the cystidia throughout length, called as ornatocystidia. And there is a lot of diversity on cystidial element. One becomes crazy looking at these cystidial structures if you have that passion for microscopic morphological characters. Next, please. The variation continues. Next one, please. It again further continues. Regularly septate cystidia. Next one. Then comes that beautiful structure, the basidia. You all have been studying fungi. We have two types of cystidia, holocystidia, which are aseptate. This group of fungi, they belong to holobasidia. Basidia will be without septa. If somehow you are getting septa, longitudinal septa, it means you are collecting a specimen other than poroid. Probably that belongs to another advanced group where there are longitudinal septa appearing in the basidia. Transverse septa, they appear in lower basidiomycota, the rus and the other lower basidia. Here, typically holobasidia. Then the second thing, number of sterigmata. Generally, in basidiomycota, there are 4 sterigmata. But here, sometimes you get 6, rarely you get 8 sterigmata per basidium. Third character, if your hyphae are clamped, basidial base will definitely be clamped. You have to ensure that. And when you see that, you will get, there is a lot of variation in shape of basidia, in size of basidia, and number of sterigmata. And believe me, all these microscopic preparations, they have been either from MSc students or from PhD students. When I was doing my PhD, the microphotography was a luxury. And we come from a state university which does not have much of funds, starved university. So we have to rely only on camera lucida drawings. And afterwards, students have started clicking, and we have now good microscopes which are available. Sir, time bata dena, jab band jata. Next one. Then there comes the basidiospores. Now, generally, there is a confusion how to ensure that this is the spore which I am observing. In Basidiomycota, spore is always attached to the sterigmata by a very small prolonged outgrowth. We call it as apiculus. Just change the focus of your microscope 2 or 3 times. Fine focusing, of course. You can see there is some kind of basal protrusion. If that apiculus is visible, it is a definite basidiospore. If that is lagging, it might be a vegetative structure. Sometimes we tap the slide badly and some fragments might be lying, which can give us an illusion of basidiospores. Now the questions, then how to ensure that we have spores preserved? There is a technique called as spore print preparation. You have collected the fresh specimen, brought it to your lab, just spread a chart paper, place a slide, clean glass slide on that, and keep your pore surface on the glass slide. Cover it with a cellophane sheet, leave it for 24 hours. The pores will release the spores, and that will be imprinted on the slide as a spore print. With a brush, take that spore print, put on the slide, and observe under the microscope. It will be a pure basidiospore preparation that will be there. So you have to look upon the shape of basidiospore, its surface. Is it smooth? Is it some kind of spiny? And thickness of the wall. Next, please. See, these are all micro photographs of the basidiospores produced in various genera of polypores. They may be ellipsoid, they may be broadly ellipsoid, they may be cylindrical, subcylindrical, allantoid. These are the 5 basic shapes. Sixth one is globose. They may be roughly rounded. Next one, please. These are all basidiospores which we could capture during the last 10-15 years at Patiala. See the variation in basidiospores. Last feature which we have to note down is the reaction of basidiospore wall. You have to put 2 stains. One is cotton blue, 1% prepared in lactophenol. So when you have seen the basidiospore, from the edge of cover slip, release a drop of 1% cotton blue. Heat your slide gently. If the wall of your basidiospore has turned bluish or blackish, we say spores are cyanophilous. If it does not, acyanophilous. Second character. There is a reagent called as Melzer's reagent. It has chloral hydrate. Now we have substitute for chloral hydrate that is available. You prepare that Melzer reagent. I will share the composition. Again, put a drop of Melzer reagent, but you need not to heat it. Leave the slide for 15 minutes. Again, observe, is there any change in the spore wall character? If it has turned darker, it is amyloid, otherwise it is anamyloid. These 2 characters are very significant for taxonomic key application. Next one, please. See, in the middle, all spores have turned black. It is a typical cyanophilous spore. Next one, please. Then you have to see the construction. Now, that is the cross section which I just mentioned in the beginning. This is how the pore will appear. Around the pore, there will be fertile layer full of basidia and cystidial elements. If you move away from the pore, in between, there is a region which is sterile region, where you will see the disappearance. You have to have in your mind what is the construction of my fructification, my specimen, how it has been organized. Both these preparations, they are seen in 1% Congo red. Congo red is a dye which is used to stain the wall of fungal structures. 1% Congo red is prepared in distilled water. Next, please. This is the basic construction, the lower zone, middle subhymenium, hymenium, and you can see the release of basidiospores on the top. This is how a fructification actually behaves in nature. And if you have noted down all these characters, next please. This is, you have to prepare some line diagrams. The traditional method is to use a camera lucida, which is with again the starved state universities. The institutes like this which encourage the research work, they can go for drawing attachment. There are few brands available in India which now give you a kind of drawing attachment. You have to put your slide under that objective, focus, the picture will be reflected on the side on your sheet of paper. You have to sit and just draw the outline projected on your sheet of paper. These are called as Olympus is there. Then I think one is by Leica. There are 2-3 drawing attachments. We are in the process of purchasing a couple of them shortly if that project matures. So you have to construct this type of line diagrams. And once this is done, your working of morphology is complete. Next, please. Then comes Next, please. Where to look for these collections? This is the kind of substrate. They grow usually on dead wood. So you have to look in the forest for these fallen logs. The decaying wood, mature forest, young forest generally has lesser number or even no fructifications. Next, please. All type of forests, gymnospermous, angiospermous, mixed forests you can get. Majority of them are present in broad-leaved angiospermous, the one like bench. Beautiful substrate for endless diversity. Next, please. Next, please. Next, please. These are the forests which we have been working for the last 15 years at Patiala. Next, please. All, even roadside, that has been exemplary for the collection of these fungi. Next, please. Dry and wet, both type of location. Next, please. So this is the basic methodology. You have to collect the specimen with the help of hammer and chisel, remove it gently along with the part of wood substrate. And then you have to go for preliminary examination, macroscopic details. Take its spore print and dry. You have to simply dry your specimens in shade. You don't require any dryer, sophisticated dryers for. No use of heater is allowed. Shade में इनको just spread it on the brown paper or newspaper sheets. Then these are the microscopic preparation grades: KOH, cotton blue, Congo red. Cotton blue and Congo red, they stain the wall of the fungal structures. Phloxin stains the contents of the fungal structure. And then Melzer reagent and sulfovanillin, they are required for reaction studies. Next, please. This is how we collect. These are all students who have been working and collecting on these specimens. Next, please. This is how we go for photography, for close-up photography. Next one, spore print. This is how it is being prepared on those glass slides. Next, please. And simply drying in shade. Next, please. And then you need a good microscope to make those microscopic preparations. Next one. And let me explain whatever we have studied by taking the example of genus Ganoderma. We have collected a specimen. It is brown, shining. We note down the basidiocarp is either sessile, subsessile or stipitate. Its attachment is either with the root or trunk. How is its upper surface? How is its pileal crust? Context, hymenophore, hyphal system, basidia. This is the rough observation sets. If you have your records on this line, your key will definitely happen if this is the case. Next, please. See, this is the variation. Next. This is the type of cuticular elements. You have to make a very thin section from one side of the pileus and look for this crust, brown crust. Either there will be a palisade depicted in first line diagram, or there will be some outgrowth, trichoderm type, or there will be anamyxoderm, mixed hyphae type. Three types of pileal crusts are present as far as genus Ganoderma is concerned. Next, please. Then upper portion context. See, there are two zones: upper light brown, lower dark brown, and then you see the tube layers. Next, please. Then you see the lower surface, its color. Next, please. Then you come to its hyphal system: binding, skeletal, generative hyphae. Cuticular elements, spores. Those are the same images of basidiospores of Ganoderma. And dear friends, if you see those ganodermoid spores, outer wall is smooth, inner wall is thick. In between the two walls, there are interwall pillars. If this is the character and one side of the spore is flat, if this kind of spore is there in your preparation, believe me, 100% the specimen is of genus Ganoderma. So only from spore character. And then next, please, you prepare a standard description. We moved from the macroscopic characters, all compiled in the form of a technical description. Next, please. Compare it with the literature. Next, please. These are the beautiful monographs published by Leif Riverden and one that is excellent contribution from India by Dr. I just forgot the name. He's from BSI Dehradun. That is the only valid monograph as far as Indian context is concerned. Next one. And you follow the key. Whatever factors we have just listed, they will definitely result and they will make it a permanent identification. Next one, please. Next one. Based on this character, we could identify 38 species of Ganoderma only from Punjab, Himachal Pradesh, and Uttarakhand. Next, please. This is the diversity, how we go for. This is the kind of pattern which we follow. Next, please. All Ganoderma. Next, please. Further it continues. You just see there are minor differences. But if they are carefully looked upon, then you can correctly identify. Otherwise, it will land you in some misidentified specimen. And recently, we started a very initial young stage study. We took some samples from market which claim to be Ganoderma lucidum products. And one of my friends, he is an expert in FTIR. And you know, with FTIR, if you have a standard profile, you can compare whether the content is actually present. And to our surprise, out of 8 samples, 6 samples did not contain Ganoderma lucidum. 2 were having, so that is the kind. This is where correct identification comes. Whatever you have picked, processed in the name of lucidum lingzi, that is not valid. Same study was conducted for Chyawanprash. We all consume it in winters. And there are 3 basic contents. This was the study which was conducted by Delhi University. And almost all samples failed in that analysis, again because of wrong identification. Next, please. Next, please. We could describe new species based on this. Next, please. I'll show you one. Again, chalo ji. Next. Next, please. There is a lot of diversity. Yes. This is the species we collected from Chandigarh. And we collected 3-4 times. Every time we ended up in a blind identification. Then we sent it to Leaf Riverdon and he authorized, yes, it is a new species. Next, please. And we could publish it in that journal, Mycotoxon, the most reputed journal in the field of mycology. This is a previous one, sir. This was a species we collected from Jammu. Very small fructifications growing on fencing pole. Based on morphology, we identified this Gloriophyllum, again a very common genus. But spores were exceptionally large. We named it as Longisporus and subjected it to the molecular phylogenetic analysis. Next, please. And that multi-gene molecular phylogeny supported our identification. Yes, it is a novel species. So if your morphology is correct, honest, dedicated, it will definitely give you wonderful results. Next, please. This is genus Tremades. Lot of diversity is there in genus Tremades. Next, please. And we could identify large number of, there is one drug called PSK. That is the most important drug, anti-cancer drug based on fungal collections. It is Tremades versicolor turkey tail fungus. And that is medicinally approved drug for cancer management. And it only comes from Tremades versicolor. Rest all are adulterations to that drug. So these are the 2 people I wish to thank. On the left, he is my mentor, my PhD guide, Professor Gurpal Singh Dhingra. On the right is the person whom we have been meeting often at Amravati, Dr. N.S. Atri. He is again from our department. So these 2 people, they really nurtured me for this kind of work. Next, please. These are people. He is Dr. Nils from Oslo University, Dr. Rivarden and Dr. Langer. Because again, sorry for repeating, we are staff university. We cannot download even the paid publications. So these are the people who come at rescue. Just be sure if you have the will to work, there is no problem in resources. Somebody somewhere will come to your rescue and will help you to overcome that particular obstacle. Next, please. Whatever I could show you is because of these 8 people. They have been working like anything. And I was blessed that 6 of them, they were the girl students. And I accompanied only 2 or 3 times for the collection. Rest all was done at their own by these girls. And the best satisfying part is all of them, they are now permanent faculties, or some of them they are abroad doing wonders in their field. Next, please. So let me thank the Almighty for that kind of blessing. And I will fail if I do not acknowledge the kind of support from both the conveners, Dr. Deshmukh, Dr. Hande, both the co-conveners, Dr. Satpute and Dr. Bobade, and the organizing secretary, Madam Mahakode. Sorry, I'm little bit uh, not very good in learning names, but gradually I will. So, and I am extremely thankful to the managing committee of Shri Shivaji Science Colleges and other colleges who have given this wonderful platform. Any questions, any queries, with the permission of Chair? Thank you.
Now the session is open for discussion.
So does the temperature affect the characteristic external color particularly?
Yes, it's a very good question. What is the role of temperature? Now, there are 3 types of collections, sir, which are occurring in polypores. We have tropical, subtropical, and temperate species. As far as temperate species are concerned, they change their color very quickly with increase of temperature. The subtropical and tropical, they are quite hardy and resistant. Because most of the subtropical and tropical collections, they are either diametic or trimetic. They are stable. Most of the temperate collections, they happen to be monometic. They are little bit unstable. So whatever collections we collect from Indian kind of thing, except for Himalaya and higher reaches of Western Ghats, they do not change their characters with temperature. They remain stable. Thank you.
Sir, apart from your morphological and anatomical identification,
Yes.
let me know about the molecular level.
Yes. As far as molecular level is concerned, if you talk about the genes which are used as barcode in case of polypores, we always start with ITS. ITS gives you a very good idea as far as the generic identification is concerned. And in the modern context, we do not go for single gene based phylogeny. So there are other markers like LSU that if the combination of ITS and LSU that has served excellent as far as our experience is concerned. Both of them, if you have that thorough regressive, one suggestion is there because in the name of sequences, lot has been deposited on NCBI. So if we have the sequences available for holotypes, that should be only taken into account. This happened with a couple of our specimens. We could not publish them because when the reviewers, they do their own blast analysis, you have to submit the NCPI files also, and then they will say it is resembling with that particular species. So holotype sequences and combination of ITS and LSU, that serves the purpose as far as polyphonics is concerned.
Wonderful presentation.
Thank you, sir. Thank you. It's all because of the blessings of Almighty and friends, and I am extremely thankful for your patience and for your time. Thank you very much.
Thank you very much, Dr. Avneet Singh, for mesmerizing talk on polyphore taxonomy. Today, when everybody is going after molecular identification, and you have given such a wonderful talk on morphological aspects, where you started with morphology and then you ended with microscopic studies. Here, the students can learn. Today, I have learned a lot how to identify the polypores and other fungi. You have simplified the topic in such a way that everybody, whosoever is now going in the field, can identify at least 2 to 3 fungi on the basis of your today's talk. So, I, on behalf of all the participants and the organizer, very much thank you. Thank you very much.
Sorry to interrupt. If there is any student who wants to come, in summer months, we have hostel available because during the session it is difficult to arrange for the accommodation. Any student, any colleague from this part wants to have some kind of training for 2-3 weeks, you can tie up. We'll be happy to cooperate. That is our mandate. We must percolate that, and you can come just giving an email. My email ID is available. Dr. Hande is there, and we can arrange for that. So at after this, thank you very much.
Thank you, sir. And now I would like to request Honorable Principal, Science College, Nagpur, Dr. Omraj Deshmukh, sir, to please take a seat on the dais. And I also would like to request Honorable Principal of Mahatma Phule College, Varud, respected Dr. Hande, sir, to please take the seat on the dais. And also our eminent speaker and resource person, respected Dr. Avneet Pal Singh, sir, I would like to request, sir, please take your seat on the dais. So it was a very good and explanatory lecture on the topic polypore taxonomy morphological approach. It is our pleasure to felicitate our chairperson, co-chairperson and resource person of technical session 1 of today's national seminar. Now I would like to request, and it is the privilege to request Dr. Omraj Deshmukh sir to felicitate chairperson Dr. N.M. Dongarwar sir, Professor Department of Botany, PGTD RTM Nagpur University Nagpur, by offering a sapling and memento. Please sir. It is my privilege to request Dr. Hande sir to felicitate Dr. G.B. Hedau sir, co-chairperson of today's technical session 1, by offering a sapling and memento. Please sir. Again, I would like to request Honorable Principal Dr. Omraj Deshmukh sir to offer a sapling to our resource person Dr. Avneet Pal Singh sir. I would like to request sir to offer a bouquet to our resource person Dr. Avneet Pal Singh sir.
Thank you so much.
Yes, thank you so much, honorable dignitaries. Now it is the time to felicitate some of our former professors and head of departments of botany from various colleges, including the Science College, Congress Nagar, Nagpur, as well as some eminent professors. So now it is my pleasure to request respected Dr. Hande sir to welcome respected Dr. R.N. Deshmukh sir, former head Department of Botany, Science College, Congress Nagar, Nagpur. Please sir. Again, I would like to request Dr. Hande sir to welcome Dr. S.A. Kalkar madam, former head Department of Botany, Institute of Science, Nagpur. Now I would like to request Dr. Omraj Deshmukh sir to welcome Dr. Dongre sir, Head Department of Botany, Mahila Mahavidyalaya, Nagpur, by offering a sapling. Again, I would like to request Dr. Omraj Deshmukh sir to welcome Dr. R.R. Khapekar sir, Head Department of Botany, Sindhu Mahavidyalaya, Nagpur, by offering a sapling. Please sir. Now I would like to request Dr. Hande sir to welcome Dr. Pohekar sir, Head Department of Botany, Institute of Science, Nagpur, by offering the sapling. Thank you so much, all the respected dignitaries. Now, for technical session 2, I hand over the mic to Dr. Zamarkar madam for further proceedings.
Thank you so much, ma'am. So we are concluding this first technical session here, and we will proceed for the technical session second. Before starting the technical session, I would like to announce that the topic for the second technical session is fungal endophytes from nature, hitchhiker to microfactors of bioactive compound. The chairperson for this session is Dr. N.H. Shahare sir. Co-chairperson is Dr. A.A. Fulzere sir. And the resource person is Dr. Vineet Meshram sir. Before starting the session, I would like to welcome all these dignitaries to please have their seat on the dais. Dr. N.S. Shahare sir, Dr. A.A. Fulzere sir, and Dr. Vineet Meshram sir. So please engross their seat on the dais, please sir. We will start first by welcoming
Yeah.
So, the first, I like to introduce our chairperson, Dr. N.S. Shah sir, our distinguished chairperson for second technical session is Dr. Narendra Haridas Shahare sir. Sir is currently serving as professor and head Department of Botany at Brijlar Biani Science College, Amravati. He has over 28 years of teaching experience and 20 years of research experience. He has successfully guided PhD and postdoctorate students, completed research projects, and published more than 30 research papers in national and international journals. Dr. Sahari sir is the author and editor of several botany textbooks, an active member of many national scientific bodies, and has significantly contributed to academics, research, and science popularization. We are honored to have him with us today. I request Dr. S.V. Satpute sir to welcome our chairperson, Dr. N.S. Shahare sir, by sapling and memento. Thank you so much, sir. Our next co-chairperson is Dr. A.A. Fulzare sir. Our Dr. Fulzare sir is currently serving as professor and head Department of Botany at Sri Mathura Das Mohta College of Science, Nagpur. He is a senior academician with over 32 years of teaching experience and 20 years of research experience in the field of botany. His area of specialization includes mycology and plant pathology. He has successfully guided PhD, MPhil, and postdoctorate students, completed major and minor research projects, and has published more than 25 research papers in reputed journals. Dr. Fulzare sir is the author of several botany textbooks, a recipient of the Young Scientist Award, and a UGC CSIR NET Fellow. He has also served as a member of boards of studies at various universities and has significantly contributed to academics and research. We are honored to have him with us today. For welcome, I invite Dr. D.V. Hande sir to welcome Dr. Fulzare sir by sapling and memento. Thank you so much, sir. Now, the resource person for the technical session second is Dr. Vineet Meshram, sir. Sir is currently working as a Dean Research and Development Cell at Anjaniya University, Nardaha, Chhattisgarh, India. Prior to this role, he worked as a visiting scientist at the Department of Plant Pathology and Weed Research, the Volcanic Institute Agricultural Research Organization, Israel, with a research and academic career spanning over 17 years. His expertise lies in the field of natural products and drug discovery, microbial secondary metabolics, microbial biochemistry, and the management of biotic and abiotic stresses in crop plants with special references to endophytic fungi. Dr. Meshram's research efforts have yielded substantial outcomes, resulting in the publication of over 25 years articles in reputable journals, along with paper presentation in over 26 national and international conferences, and contributions to 5 book chapters. So, served as a reviewer and editor for numerous Eastern journals. He also holds 4 patents to this name. His contributions have been recognized through notable awards. The Mycological Society of India awarded him the Young Scientist Award 2012 and 2014, and European Mycological Society the same honor upon him in 2015. Additionally, he received the Carson's Award in 2019 for the Ministry of Agriculture, Agricultural Research Organization in Israel, in appreciation of his investigations into forestry research. Recently, he was awarded with Professor B.P.R. Vittal Memorial Award by the Mycological Society of India. I invite Dr. D.V. Hande sir to please welcome our resource person Dr. Vineet Meshram sir with sapling. Thank you so much, sir. For the further proceeding, of technical section second, I hand over mic to chairperson Dr. N.H. Sahare sir.
It's a great privilege to stand in this auspicious college auditorium because it was founded by a well-known educationist, Punjab Deshmukh sir. Respected principal sir, the senior research professors, my future colleagues, and invited speakers. Now, already the biodata of the very well-learned today's speaker, second speaker, Vineet Meshram, sir, already we are here. Now, without wasting too much time, I will request him to start his talk. Thank you.
A very good afternoon. Before I start, my sadar naman to Jyotibai and Savitribai Phule and Bharat Ratna Shri Baba Saheb Ambedkar, to the great visionary Tukreji Pitraji Maharaj and Panjabrao Deshmukh. I thank the organizers, especially convener Dr. Khande and all the members, Dr. Deshmukh and others, for inviting me and giving me this opportunity to present my work before you. With the permission of the chair, is it okay if I speak in English? Mix of Hindi plus English? Because being a Mishram, माझे मराठी येत नाही. That's a bad part of me. I'm trying to learn. And if I speak a Marathi mixed with Chhattisgarhi, I think it will not be good. Yeah. So I'll try to speak. Is it visible to you, students? Okay. So I'll be speaking with endophytes. Before I start, I would like to deal with his story. And somewhere around 7th century, there was a lot going on in a country now known in the Middle East, and some of the peoples that came and ran away from there and came to a port in Gujarat called as Sajan. And the prince who was there, उन्होंने कहा कि हम आपको आश्रय नहीं दे सकते, we are already loaded with it. So how can you accommodate you when we don't have the resources? So one of the learned member, he took the milk, put some sugar, and mix it. And see, sir, we will mix you just like the sugar mixed with the milk. Those community, we know them as Parsis in India, and we all know what contribution they have in the development of Indian society. Just like what we cannot see the sugar mixed in the milk, there is a group of fungi that we call endophytes, which are within the plants and cannot be seen without, because they don't show any kind of symptoms, no negative effects, just good things. And these endophytes provide different kinds of functions. All good things you are going to hear, whether it's going to be in relation to the industries, in relation to the pharmaceutics, in relation to the agriculture. So one by one, we'll go and we'll see what endophytes are. So as the name suggests, endophytes are something that are ubiquitous in nature. Ubiquitous means omnipresent and polyphyletic. A different range of phylums they have been found. So when I say move, then only move. Okay. That are found inside the plant or a portion of their life cycle. They uh, live inside the plant without showing any negative impact. So if you are seeing a plant composed of three types of microflora. One is pathogenic, you see the symptoms. One is saprophytic, that are present on the surface. And the third one that we are talking about is the endophytes, which which resides within the plants without showing any negative impact. So, as we break the name, endo means inside and phyte means plant. So, any organism, be it fungi, bacteria, viruses, or any microorganisms that live within the plant and do not cause, while living there, any kind of negative or pathogenic interaction, that are known as endophytes. Next, please. So, if you take a plant, it is present in all the tissues, whether it is ribospheric or phylospheric. So, from roots to stems to leaves to flowers to seed, you will find endophytes in all the areas, and you can see they are present in the intercellular, intracellular, and intracellular spaces of the cells, and they colonize all these without showing any kind of symptoms of their present. What they do, next please, what they do, because they are living with the constant interaction with the plant, they are providing them health benefits. भाई अगर रहना है साथ में तो कुछ ना कुछ तो करना ही पड़ेगा, है ना? This world is all about give and take. Because endophytes and fungi cannot produce their own food. So what they do? They colonize this plant and they take the food from them, and in reward, what they do, they provide certain health benefits, like they provide resistance against biotic and abiotic stress. For example, आप लोगों ने हाईवेज देखे होंगे, हाईवे के किनारे हमेशा कनेर के प्लांट होते, क्यों? Somebody thought of it? Because it produces certain kinds of phytochemicals or scent that is anti-grazing. Herbivores को बाहर भगाता है. And research has found that it is because of the endophytes that are present there, they are producing these phytochemicals which are anti-gazing. So that's why. So this is one of the thing you can see that what endophyte is doing. So there are continuous endophyte and plant communication, and this leads to what? Next please. Next please. So this endophyte, what it does, it says it is in continuous exchange with the plant. There is exchange of genetic material through vertical and horizontal transfer. Also, there is change of metabolites. Okay, and there is one hypothesis that is known as hormosis, which means whenever the plant there is any kind of biotic or abiotic, this endophyte, it senses it and it releases phytochemicals or different kinds of chemicals, जिससे कि क्या होता है कि प्लांट्स को एबायोटिक एंड बायोटिक स्ट्रेस को मिटिगेट करने की प्रॉपर्टीज मिलती है. नेक्स्ट प्लीज. So when we classify endophytes based on their origin and habitat, there are two types of endophytes, well defined by Rodriguez in 2009, published in New Phytologist. They are Clavicipedius and non-Clavicipedius. So first type is class 1 Clavicipedius, that is usually the root fungi that found in the grasses. Okay? कई बार क्या होता था कि there were in Europe that there were deer grass और केटल्स जब उसकी ग्रेजिंग करते थे तो मर जाते थे. तो फिर रिसर्च में ये पता चला कि what is the reason, and reason because endophytes were producing inhibiting there and they were producing toxic phytochemicals, anti-gazing phytochemicals, जिसके कारण वो मर जाते थे. So type 1 is Clavicipedius, which is usually in grasses, and then non-Clavicipedius, इनके 3 subtypes है, type class 2, class 3 and class 4. I usually work with class 2 because they are found in root, leaf, stems, all this, and they are habitat adapted. Habitat adapted मतलब if suppose there is a plant growing in Sahara desert or Thar desert, बहुत ज्यादा वहां पे dry nature है, but you'll find some plants that are absolutely green. वो इसलिए होता है because of the microbiome that is present inside, which is allowing it to maintain the moisture and the other things inside the plant. So class 2, then class 3 and class 4, they are only found, class 3 is only found in leaves, class 4 is only found in the rhizome. So class 2 we will be discussing, that is from root, leaves and stems, and that are vertically or horizontally transferred. मतलब कि सोर्स से भी हो सकते हैं and also transferred from the roots. Next please. So endophytes as microfactories. हुआ क्या कि one of the very deadly disease which we are facing here is cancer. Most of the death after the cardiovascular diseases is cancer. Okay, and remedy to cancer or a drug to cancer is Taxol. But can anybody tell कि Taxol आता कहाँ से? Texas batata. So for one injection, you need 65-year-old Texas plant to be processed to produce 10 mg of an injection. हो यही था ओवर एक्सप्लॉइटेशन और इतना एक्सप्लॉइटेशन हुआ कि टेक्सास का जो प्लांट है नाउ इट इज इन एंडेंजर्ड कैटेगरी. So in 1990s, 91, 92, NIH, National Institute of Health, USA, इन्होंने एक प्रोग्राम चलाया. They asked a team of researchers headed by Professor Gary Strobel, you find an alternative. क्या होगा? So what they did, they came and took the Himalayan yew tree. वहां से उन्होंने एंडोफाइट्स निकाला एंड फाउंड देयर इज वन पर्टिकुलर एंडोफाइट that is Exomyces endrinae, which is producing the exact taxol that was produced by the host plant. From there, it was the endophytic research was revived, or you can say it's a renaissance of endophytic research. वहां से लोगों ने एंडोफाइट्स को टेक आउट किया एंड फ्रॉम टेक्सल आज की डेट में यू कैन सी अराउंड 10 to 20,000 endophyte plant similar products reported by endophytes. Next, please. Next. So you can see what endophytes can produce. Taxol, the anti-cancer drug. Photophylotoxin, another anti-cancer drug. Camptothecin, another anti-cancer drug. Huperazine, used for Alzheimer's disease. Vincristine and vinblastine, the vinca alkaloids, also anti-cancer drugs. Curcumin, the हल्दी हम रोज दूध के साथ लेते हैं. Curcumin is there, and the resveratrol. Girls might be interested in resveratrol. You know why? Anti-aging, anti-acne. Okay, so and there is a French paradox. Why? Anybody knows about the French paradox? फ्रांस के लोग जो हैं वो बहुत ज्यादा चीज और फैट खाते हैं, फिर भी उन्हें सबसे कम हार्ट डिजीजेस होते हैं. कारण है क्योंकि वो वाइन बहुत पीते हैं, और वाइन में कंपाउंड होता है रेड वाइन में which is resveratrol, which protects them from heart disease. This is known as French paradox. So you can see there are different kinds of bioactive compounds that are present within the endophytic fungi. So one by one we will see how you isolate, how you collect, how you isolate, and how you explore the endophytic fungi. Next, please. So plant sample collection. As we discussed, जब भी हम plants लेंगे तो तीन चीजें याद रखेंगे. सबसे पहले that we have to remove the pathogenic part. So only the healthy and the mature plants samples will take, which are very fresh. Okay, so आपने pathogenic हटा दिया, अब बचे आपके पास endophyte and saprophyte. Saprophyte के लिए क्या करेंगे? You will do the surface sterilization. Okay, so we'll see how. And जितनी भी less polluted जगह में आप जाएंगे, the more you go to the dense forest, and we are blessed कि हमारे पास इतना dense forest है यहाँ पे, है ना? So we can go to the dense forest. The rainforests are considered as the most prolific sources of endophytic fungi that are having biological activities. Next, please. So what we do, we chose two biodiversity hotspots. One was the Western Ghats, and one was the Northeast Himalayan region. And with the blessings of Lord Buddha, that's me, some ages before, when I didn't have a tummy. So that was me in 2010, and we went to the Tiger Hills in Darjeeling, and we collected this is Cinnamon camphora plant. जिससे कपूर बनता है. Okay, and we collected different plants from Cinnamon camphora to Cinnamon zelenicum, Cinnamon mulbericum, also Moraia, Catharines roseus, Tabernamontana diabericatia. From these two different plants, one is the Western Ghats, Kerala and Karnataka, and one is the northeastern parts. Best practice ये होती है कि जैसे ही आप प्लांट कलेक्ट करते हैं, within 48 hours आपको अपने सेंटर में आके उसको प्रोसेस कर देना है. अगर नहीं कर पा रहे हैं तो वहीं के वहीं प्लांट को पहले अच्छे से डिस्टिल्ड वाटर से या टैप वाटर से धोइए और उसको 30% अल्कोहल से हल्का सा रब कर दीजिए. तो क्या होगा, जो भी एपिफिटिक फंजाई होगी उसके ऊपर, दैट विल बी वॉश्ड ऑफ. देन रैप इट विथ अ स्टेराइल टिश्यू पेपर और उसको जिप पाउच में बंद करिए. मार्किंग करिए कहां से आपने लाया है, नेम, लोकेशन, डेट, वेरी इंपॉर्टेंट. और ये नहीं कि पूरी की पूरी टंगियां ही काट के ले आए. मैक्सिमम 5 लीव्स आर इनफ. 5 लीव्स, थोड़े बहुत stems and other parts, whatever you want to take. Next, please. So these are the samples that we have collected. This is Cinnamon mulbericum. The next one is Zelanicum. Third one is the Camphora. Fourth one is Agel marvelous, one of the best plant to study endophyte. Whatever the best result that I have got is from Agel marvelous. और हमारे यहाँ तो कहा ही जाता है कि बेल पत्री जो है उसका जो फ्रूट है या लीव्स है इज वेरी गुड फॉर डायबिटीज, है ना. डायबिटीज लोगों को कहा जाता है कि उसके लीव्स को बॉईल करके पीजिए. ओके, सो ट्रेडिशनल नॉलेज टू modern knowledge. Agel marvelous, then Tabernae montana divaricata and Catranthus roseus. And Catranthus roseus gave me my PhD fungi, which we worked on cardiovascular disorders. We'll discuss it later. So now the plant is or the samples are collected. Next please. अब हम इसको प्रोसेस करना शुरू करेंगे. नेक्स्ट, सो द सैंपल इज कलेक्टेड, ब्रिंग टू योर लैब. अब हमको ऊपर का जो भी गंदगी है या एपिफाइट्स है उसको हटाना है. तो क्या करेंगे, सबसे पहले 1% सोडियम हाइपोक्लोराइट में उसको इमर्स करिए इनसाइड अ लेमिनार फॉर 3 to 4 मिनट्स. आगे पीछे जैसे आप पापड़ घुसते हैं बिल्कुल उसी के तरह हाइपोक्लोराइट में लीजिए और उसको अच्छे से इमर्स करके आगे पीछे साफ करिए. 3 मिनट के बाद 70% इथेनॉल फॉर 1 मिनट, देन 30% इथेनॉल फॉर 45 सेकेंड्स. आपका स्टरलाइजेशन इज डन. अलाउ इट टू ड्राई इनसाइड द लेमिनार. सारा पानी इवेपोरेट हो जाएगा. जैसे ही पानी इवेपोरेट हो गया, कट इनटू वेरी स्मॉल पीसेस. नेक्स्ट जाएंगे क्या सर? Cut into very small pieces, not more than 3 to 5 centimeters. 5 centimeters too much. 2 to 3 centimeters are more than enough. कट करिए और पीडीए प्लेट्स में लगा दीजिए. So this is what. These are the plates. These are the cut sections. This is stem. This is seed. और आप देखेंगे कि यहाँ पे जो है the fungi started growing after 3 to 4 days. यहाँ पे देखिए within the plants. Okay. Or now you can purify it. Once the purification is done, what you will do, purify करिए और purify करके इसको store कर दीजिए. store करने का सबसे initial तरीका है prepare PDS lands. PDS lands के बाद उसको 15% glycerol solution में डाल दीजिए और -80 डिग्री में 3 से 4 साल के लिए आप रख सकते हैं. So following this process, we have isolated somewhere around 700 fungal isolates from different plant species. Next please. And you can see how beautiful fungi we have seen obtained from this collection. So the first one, this is Fusarium incarnatum. This is Fusarium solani. This is Fusarium equisetae. This is Lesodiclodia. This is again Fusarium oxysporum. Fusarium equisetae. This is Polytotrichum. Alternaria, Fusarium, Trichoderma, Penicillin, Bioactive, Lesodiclodia, and that one is the Pestiolopsis species. Once you get that, अब मूव ऑन होते हैं हम कि इसका माइक्रोस्कोपिक आइडेंटिफिकेशन कैसे करेंगे. नेक्स्ट प्लीज. तो सबसे सिंपल है, नेवर पुट योर एंडोफाइट ऑन पीडीए. पीडीए, आई डोंट नो आई शुड से दिस और नॉट, बट पीडीए इज द वर्स्ट मीडियम. इतना ज्यादा शुगर है, जैसे ही शुगर होगा वो सिर्फ माइसेरियम देगा और हमको तो स्पोर चाहिए. स्पोर मिलेगा कैसे? भूखा, भूख से मार डालो. तो बेस्ट थिंग इज पानी लीजिए जो नल का पानी आता है और 2% डाला उसमें ग्रो करिए, गेट ब्यूटीफुल स्पोर्स. एंड यू कैन सी फर्स्ट टू आर Fusarium equisetae. The next two are Lesodiplodia. One is the immature and the second one is the mature conidia. Third one is the third one is the Curvularia. Okay, third cell is curved and elongated. The F1 is Fusarium oxysporum. Fusarium equisetae only produces macroconidia. It doesn't produces microconidia, whereas Fusarium oxysporum it produces macro and microconidia. उसके बाद then there is Alternaria, the next one is Bionectaria, and the last one is Pestiolopsis species. So you can just by microscopic evaluation you can see the endophytes in a very good way. But as sir rightly said, आपको माइक्रोस्कोप में टाइम बिताना पड़ेगा, वो बेसिस है. जब तक आप टेक्सोनॉमी नहीं करेंगे, एंड आई एम टॉकिंग टू द यंग रिसर्चर्स जो कि अब माइकोलॉजी पढ़ेंगे, जब तक आप माइक्रोस्कोप में बैठ के टाइम नहीं बिताएंगे, इतना टाइम बिताइए कि आपको प्यार हो जाना चाहिए अपनी एंडोफाइट से, सोते जागते वही दिखना चाहिए, ठीक है। तो एंडोफाइट्स आइडेंटिफाइड, अब हमारे पास एक बेसिक है। अब क्या कर सकते हैं आप, क्योंकि आपको पता है कि मैंने इस प्लांट से ये एंडोफाइट लिया है, सो यू कैन गो टू द लिटरेचर एंड सी कि व्हाट आर द प्रॉपर्टीज देयर है, और उसके हिसाब से आप क्या कर सकते हैं, उसको एक्सप्लोर कर सकते हैं। नेक्स्ट प्लीज, नेक्स्ट करते रहिए, करिए और करिए। तो सबसे इंपॉर्टेंट चीज हो गई स्क्रीनिंग होती है ना, कि आप स्क्रीनिंग कैसे करेंगे। स्क्रीनिंग के लिए क्या करेंगे, सबसे पहले कल्चर फिल्ट्रेट प्रोडक्शन होगा। कल्चर फिल्ट्रेट प्रोडक्शन के लिए या तो आप पीडीबी यूज कर सकते हैं, पोटैटो डेक्सट्रोज ब्रॉथ, या तो आप जेपेडॉक्स मीडियम यूज कर सकते हैं, या कोई भी मिनिमल मीडिया अकॉर्डिंग टू योर नीड। उसको शेकिंग इंक्यूबेटर में 120 to 150 आरपीएम फॉर, इफ यू आर यूजिंग 100 ml, 10 days इज इनफ। आपको फिर भी लगता है कि थोड़ा लेट ग्रोइंग है, क्योंकि एंडोफाइट्स बहुत स्लो ग्रोइंग होते हैं। अच्छा एंडोफाइट वही है जो बिल्कुल छोटा सा होगा और बहुत स्लो ग्रोइंग होगा, अदरवाइज तो फिर सूडो एंडोफाइट्स आर फास्ट ग्रोइंग। ओके, तो एंडोफाइट्स लीजिए, उसको ग्रो करिए, अब उसको फिल्टर आउट करिए, माइसीलिया अलग और ब्रॉथ अलग। अब इसमें आप यू कैन चूज 2 वेज। आपको इंटरसेलुलर देखना है तो क्रश योर माइसीलिया विथ कोल्ड मिथेनॉल। कोल्ड मिथेनॉल क्यों, क्योंकि ठंडा मिथेनॉल जैसे ही जाएगा और क्रश करेंगे, सेल वॉल ब्रेक होगी और सारा का सारा मेटाबोलाइट बाहर आ जाएगा। ओके, और गो फॉर द सॉल्वेंट एक्सट्रैक्शन। सॉल्वेंट एक्सट्रैक्शन में क्या करेंगे, वी चूज different solvent based on the polarity, so you can grow from non-polar to polar or polar to non-polar depending on the choice. Okay, so what you do, आप इस तरीके का सेपरेटिंग फनल लेंगे, सबसे इंपॉर्टेंट है ट्राइपॉड के नीचे बीकर रखिए और इसको बंद करिए, नहीं तो मोस्ट ऑफ द टाइम क्या होता है, खुला रहता है नीचे से बह जाए, तो आपकी 15 दिन की मेहनत चले जाएगी। तो मीडिया डालिए, जैसे ही you put media, add twice the amount of or thrice the amount of solvent। अगर आप 100 ml का मीडिया ले रहे हैं तो उसमें कम से कम 300 ml इथाइल एसीटेट डालिए और कवर करिए और उसको शेक करना चालू करिए। दिस वी विल डू इन द वर्कशॉप टुडे। ओके, उसको शेक करना चालू करिए, तब तक शेक करिए जब तक आपके हाथ दुख ना जाए। ट्राइपॉड के ऊपर रख दीजिए और लेट इट सेटल डाउन। तो आप देखेंगे कि एक लेयर नीचे है, एक लेयर ऊपर है, और वैसे वर्सा बेस्ड ऑन द मॉलिक्यूलर वेट। ठीक है, जैसे जैसे ये होते जाएगा आपको कम से कम 15 से 20 मिनट की एक्सरसाइज करनी है। उसके बाद क्या करिए, मीडियम को अलग निकाल लीजिए और एक्सट्रैक्ट को अलग निकाल दीजिए। अब आपका जो एक्सट्रैक्ट है देखिए वो अलग निकल गया। ओके, उस एक्सट्रैक्ट को कीप इट इन 37 डिग्री इन इंक्यूबेटर फॉर इवेपोरेटिंग। तो क्या होगा, बीकर में सारा इथेनॉल, सारा जो सॉल्वेंट है वो क्या हो जाएगा, इट विल बी इवेपोरेटेड एंड व्हाट विल बी लेफ्ट इज योर बायोएक्टिव कंपाउंड्स। ओके, फिर क्या करेंगे, अब आपका इथाइल एसीटेट लेयर आ गया। अब आपके पास जो मीडियम बच गया है उसको फिर से डालिए सेपरेटिंग फनल में, एंड व्हाट यू डू, गो टू द नेक्स्ट सॉल्वेंट, दैट माइट बी क्लोरोफॉर्म। क्लोरोफॉर्म में लीजिए, फिर एक्सट्रैक्ट करिए, फिर सेम प्रोसीजर। जैसे ही आपका एक्सट्रैक्ट आने लगे, डिसॉल्व इट ईदर इन मिथेनॉल और डीएमएसओ और सॉल्वेंट ऑफ योर चॉइस। मिथेनॉल इज द इजीएस्ट बिकॉज इट्स अ यूनिवर्सल सॉल्वेंट, हर चीज डिसॉल्व कर देता है। अगर आप नॉन पोलर लेंगे तो सिर्फ नॉन पोलर पार्ट को लेगा, पोलर लेंगे तो इट विल ओनली टेक द पोलर पार्ट। सो बेटर गो फॉर मिथेनॉल। जैसे ही आप मिथेनॉल लेंगे इन अ रिक्वायर्ड क्वांटिटी, जैसे हम यूजुअली 1 mg पर ml का स्टॉक बनाते हैं, आप 5 mg भी बना सकते हैं, 10 mg भी बना सकते हैं, अकॉर्डिंग टू योर चॉइस। नाउ यू हैव द बायोएक्टिव कंपाउंड और द कंपाउंड ऑफ योर इंटरेस्ट व्हिच यू वांट टू इवेलुएट. सो अब देखते हैं कि इवेलुएशन कैसे होंगे. नेक्स्ट प्लीज. सो लेट्स स्टार्ट विद द बेसिक. हमें करना है एंटीमाइक्रोबियल टेस्टिंग. Antimicrobial testing can be done by two ways. One is the diffusion, another one is the dilution method. Diffusions के दो methods होते हैं. One is the disk method, which is known as the KB or the Kirby-Borer disk method. Second one is the agar-well diffusion method, and third one is the dilution method that we use for obtaining the minimal inhibitory concentration or the MIC. So we'll see one by one. For doing antimicrobial activity, one thing is very important. Always follow CLSI guidelines. आप अपने मन से एंटीमाइक्रोबियल एक्टिविटी नहीं लगा सकते. क्यों? क्योंकि you need to follow certain protocol to do that. Okay? So for diffusion, one thing is very important. You need a diffusing enhancing media, and that is MH media, Muller-Hinton media. Always use Muller-Hinton media for going for antibacterial activity. अगर आपको स्टेफिलोकोकस ऑरियस, स्ट्रेप्टोकोकाई, क्लेब्सियला, ई कोलाई इनके अगेंस्ट अगर टेस्टिंग करनी है तो आप यूज करेंगे मुलर-हिंटन अगार. अगर आपको कैंडिडा के अगेंस्ट यूज करना है तो यू विल ऑलवेज यूज सेबियो डेक्स्ट्रोज अगार, SDA. ओके, तो आपने मीडिया बनाया. मीडिया बनाने के बाद उसकी जो थिकनेस होनी चाहिए 90 mm प्लेट्स में, वो 4 mm होनी चाहिए, 4 to 5 mm. अब ये कैसे करेंगे? So in a 90 mm plate, always pour around 22 ml of media. वो आपको सूखने के बाद 4 mm की थिकनेस देगा. This is standard plate size of CLSA. ओके, तो मीडिया बन गया, आपने प्लेटिंग कर दी. प्लेट करने के बाद you can load your bioactive compounds on the this that you are having, allow it to dry, and then transfer it to the medium. Next time here, and you can see around the this you'll find zone formation. Okay, this zone formation you can see in the ethyl acetate fraction there is zone formation. In the aqueous section जो बच जाता है बाद में ब्रॉथ का, उसमें कोई भी नहीं है. कंट्रोल इज नॉट शोइंग एनी infection. This is again Staphylococcus aureus clinical isolate that we got from Rajendra College, Patiala, sir. So we checked it using that. Always do testing screening 1, then confirmatory in triplicate, and always repeat your experiment twice. तभी वो confirmatory है, तभी आपको सही standard deviation मिलेगा. Okay, so once KB is done, move to next one. And this is the more preferred one, agar-well diffusion ऐसे, क्योंकि क्या होगा, डायरेक्ट जो है वो वेल में आपका कंटेंट इट विल डिफ्यूज बेटर. ओके, डिफ्यूजन बेटर करने के लिए यू कैन ऑलवेज यूज DMSO. थोड़ा सा DMSO अपने बायोएक्टिव कंपाउंड के साथ मिला लीजिए. तो DMSO क्या होता है, ज्यादा बेटर, इट enhances the diffusion, so you get better antibacterial activity. ओके, तो क्या करेंगे, सेम MHA प्लेट्स बनाएंगे. MHA प्लेट्स बनाने के बाद व्हाट यू कैन डू, यू कैन स्वैब योर बैक्टीरिया. बैक्टीरिया का काउंट होना चाहिए 1 million cells per ml. उसके लिए एक स्टैंडर्ड होता है 0.5 मैकफार्लैंड, जो कि आप एक सॉल्यूशन है बना सकते हैं. यूजुअली उसकी कंपोजिशन नेट में आपको मिल जाएगी. उससे टर्बिडिटी चेक करिए. अगर नहीं कर पाते तो आप क्या करिए, सिंपल जो सेल्स हैं उसको डिस्टिल वाटर या NaCl में डिसॉल्व करिए और उसकी OD ले लीजिए. इफ द OD कम्स टू 0.1, that is equivalent to 10 to the power 6 cells per ml. Now it is ready for swabbing. कॉटन लीजिए, कॉटन में like this, और फिर एक पोर्शन स्वैब. Suppose this is a petri plate. Okay, swab it like this, roll it, swab it like this, again roll it, swab it like this. 3 बार कर दिया. Once it is done, leave it for 20 minutes. सारा अंदर आ जाए. Okay, सेटल हो जाए. जैसे ही सेटल हो जाएगा, what you do, you punch different wells. हमने wells punch किया. एक कंट्रोल रखिए, 3 जो है उसमें अपने कंपाउंड्स डाल दीजिए। तो आप देखेंगे that how beautiful it diffuses after 37 hours. ओवरनाइट रख दीजिए, नेक्स्ट डे आएंगे 37 डिग्री में, जैसे ही आप प्लेट निकालेंगे यू विल गेट ब्यूटीफुल जोन्स अराउंड द वेल। हमेशा 3 कैटेगरीज में बांटे जाते हैं जोन साइज को। वन इज योर वीक, दैट इज बिलो 10 mm. One is intermediate, 10 to 15 or 18 mm, and one is strong, that is above 18 mm. अगर इससे ज्यादा का जोन आ रहा है तो आप इन 3 कैटेगरीज में बांट के यूजुअली अपने बैक्टीरिया कंपाउंड्स को चेक कर सकते हैं। ओके, इसके बेसिस पे यू कैन सी विच ऑफ द बैक्टीरिया कंपाउंड इज एक्चुअली ऑफ योर कंसर्न और जिसको आप आगे लेके जा सकते हैं। ओके, नाउ नेक्स्ट प्लीज, we are moving to the MIC, dilution method. Once it is confirmed that out of 100 endophytes or 100 fractions of endophytes, 10 fractions are active that you really want to pursue. उन 10 fractions को अब क्या करना है, हमको मिक निकालना है। मिक इज द मिनिमल इनहिबिटरी कंसंट्रेशन जिसमें की डेथ हो जाती है सेल्स की। ओके, तो आप क्या करिए, 1 ml 1 mg per ml का स्टॉक बनाइए और उसका 2 फोल्ड डाइल्यूशन बनाते जाइए। 1 mg per ml, 0.5 mg per ml, 0.25 mg per ml, 0.0625 mg per ml, ऐसा स्टॉक बनाते जाइए। मिनिमम ऑफ 10 डाइल्यूशन बना लेंगे और उसके बाद जो है उसको क्या करेंगे, यूज करेंगे। तो हाउ वी यूज इट, वी यूज इट इन अ 96 टाइटर प्लेट। अगर आपके पास नहीं है 96 टाइटर प्लेट, यूज इट इन एपेंडॉफ, अदरवाइज यूज इट इन टेस्ट ट्यूब्स, रिजल्ट्स आएंगे। ओके, तो जैसे ही आप क्या करेंगे कि मीडिया डालेंगे, मीडिया कैन बी MHB, मुलर हिंटन ब्रॉथ, और सेबियोडेक्स्ट्रोज ब्रॉथ, वही 1 मिलियन सेल्स, उसका हमने लिया 120 माइक्रोलीटर, 120 माइक्रोलीटर ऑफ मिलर हिंटन ब्रॉथ। उसके बाद हमने उसमें कल्चर डाल दिया। सपोज करिए आपको स्टेफिलोकोकस ऑरियस के अगेंस्ट यू वांट टू सी, सो हमने कल्चर डाल दिया एंड वी लेफ्ट इट फॉर 3 इयर्स ताकि वो ग्रो हो सके। आफ्टर 3 इयर्स क्या करिए, आप अपना जो एक्सट्रैक्ट है उसका 30 माइक्रोलीटर डाल दीजिए। 30 माइक्रोलीटर जैसे ही डालेंगे उसके बाद उसको रख दीजिए फॉर 37 at 37 degree for overnight. Next day आइए, take out the plate, inside a laminar add tetrazolium salt. Tetrazolium salt क्या करेगा, लाइव सेल्स के साथ रिएक्ट करेगा and it will give either a pink or a purple color. Okay, so रखिए, 1 से 1.5 घंटे में उसका कलर आ जाता है टेट्राजोलियम सॉल्ट का। ओके, so where the live cells will be there, the color will be violet, और जहां डेड सेल्स होंगी वहां पे कोई कलर चेंज नहीं होगा। सो यू कैन सी दिस इज अ 96 well plate, different dilutions, okay, and in triplicates 1 2 3 1 2 3 1 2 3 ऐसे triplicates में हमने किया हुआ है। ओके, सो एट दिस, फ्रॉम दिस कंसंट्रेशन टू दिस कंसंट्रेशन, मतलब 150 माइक्रोमोलर टू 10 माइक्रोमोलर, देयर इज नो ग्रोथ। अब उसके बाद ग्रोथ आ रही, so your mix is here F, and what is F? 5 micromolar concentration. At this much less concentration you are getting activity against Staphylococcus aureus, E. coli, and Pseudomonas aeruginosa. So this is how you do your minimal inhibitory concentration. इसके आगे जब आपके पास डेटा है, you can also use various statistical tool और उससे IC50 निकालेंगे। IC50 and IC90 are something that inhibitory 50% and inhibitory 90%. Okay, that you can use graph pad or other tools जिसके थ्रू वो निकल जाता है। Next please. So antioxidant के बाद another activity that we see is antioxidant activity, and on antioxidant activities के लिए 2 एसिड यूज होते हैं, one is DPPH and another one is FRAPS, ferric reducing. Okay, so करना कुछ नहीं है, आपका जो एंटी माइक्रोबियल कंपाउंड, जो आपका बायोएक्टिव कंपाउंड है, टेक इट इन अ एपेंडॉफ, मिक्स इट विद DPPH, कीप इट इन डार्क फॉर 30 मिनट्स, वो आपको कलर चेंज विजिबल हो जाएगा। If the color change is visible, if it is reducing, you know that it is antioxidant in nature. Next आइए, So we did with our endophytes, and you can see with chloroform extract, ethyl acetate extract, and methanol extract. Next, please. All these are showing very good antioxidant activity. Next, please. And we have shown here 5 of the most potent isolates, which are Phyllostica, Diporthic, Polytotrichum, and Nigrospora species, which are showing above 50 to 60% of the reducing activity. Okay, once the antioxidant activity is done, you can also go for 3 more types of antioxidant activity. One is the catalase activity. Catalase activity is the easiest but very difficult to perform. क्योंकि इतने जल्दी reactions होते हैं कि you cannot take the reading. Okay, so catalase में nothing, you have to add catalase, you have to add sulfuric acid to it. Okay, वो bubbling दिख जाती है आपको. Okay, then peroxidase activity होती है glycol peroxidase, and the third one is superoxide dismutase activity, which you can see जिसमें violet color जो होता है वो change हो जाता है. But if you are looking for only in prospect of the pharmaceutics, just look into DPPH and FRAP acid. That are enough. इसके अलावा आप SOD एक्टिविटी, सुपरऑक्साइड डिस्म्यूटेज एक्टिविटी कर सकते हैं. ओके, क्योंकि जैसे जैसे बॉडी में एंटीऑक्सीडेंट्स बढ़ेंगे, रॉस बढ़ेगा, और जैसे जैसे रॉस बढ़ेगा वैसे वैसे बॉडी में म्यूटेशंस होना चालू हो जाएंगे, और जैसे जैसे म्यूटेशंस होते हैं देयर आर चांसेस ऑफ कैंसर्स टू बी गेटिंग. इसीलिए बेस्ट ऑफ द एंटीऑक्सीडेंट को बता सकते हैं कहां होता है? वेरी नेचुरल, इट इज फाउंड, हर घर में होता है. स्पेशली टू द स्टूडेंट्स, मंदिर जाते हैं आप लोग तो सुबह तुलसी के पत्ते के साथ पानी देते हैं, ठीक है? यूनिवर्सल सॉल्वेंट वाटर, वो तुलसी से एक्सट्रैक्ट करता है, एंड इट कंटेन्स मोस्ट ऑफ द एंटीऑक्सीडेंट्स. दिस इज अ प्रूवन रिसर्च, सो आई कैन से दैट. ओके, ओके. आफ्टर एंटीऑक्सीडेंट, what we can do next, please? We can go for anti-cancer activity. Anti-cancer activity दुनिया में कार्डियोवास्कुलर डिसऑर्डर्स के बाद सेकेंड मोस्ट मॉर्टेलिटी जो होती है वो एंटी कैंसर से होती है, स्पेशली ब्रेस्ट कैंसर से फॉर विमेन्स. ओके, सो प्रॉपर स्क्रीनिंग शुड बी डन. ओके, और अब तो पेप्लो वायरस के अगेंस्ट वैक्सीन भी आ गई है, सो फॉर ऑल द यंग विमेन्स प्लीज वो वैक्सीन लीजिए. आपको ओवेरियन कैंसर से वो बचाता है. एक 10,000 की वैक्सीन है और आप लाइफ लॉन्ग सेफ है उसके लिए. 35 साल से पहले अगर आप ले लेते हैं तो यू आर सेफ इनफ. Okay, or maybe in next few years, it is in proposal with the government of India. वो जैसे आपको पोलियो वैक्सीन लगती है वैसे ही वो वैक्सीन भी लगने लग जाएगा. Okay, so what we did anti-cancer activity. Anti-cancer activity is also the same जैसे आप मिक निकालते हैं, बिल्कुल सेम है. सबसे पहले हमने 3 कल्चर्स लिए. वन इज हेला, दैट इज सर्वाइकल कैंसर. सेकंड इज द ब्रेस्ट कैंसर, एंड थर्ड वन इज द metastatic cancer which is resistant to cisplatin and other drugs. Okay, so क्या किया? सबसे पहले वही माइक्रोटाइटर प्लेट, सेम कंसंट्रेशन 1 मिलियन सेल्स को सीड कर दिया. सीड करने के बाद हमने उसको 24 से 48 आवर्स के लिए ग्रो करवाया. Once it is grown, we add our bioactive compound and let it grow again for 24 to 48 hours. उसके बाद again we add टेट्राजोलियम सॉल्ट. सेम रिएक्शन होगा. जहां लाइव सेल्स होंगी वहां पे कलर चेंज दिखेगा, जहां लाइव सेल्स नहीं होंगी वहां पे कलर चेंज नहीं दिखेगा. ओके, नेक्स्ट प्लीज. नेक्स्ट. So you can see we have taken 3 cells, and you can see at very low concentration, somewhere around 30 to 40 μg, it is giving 50% of inhibitions of the cancer cells. Next please. And you can see this is the control cell with most of the cells, and the treated one you can see, especially with MDA MB, you can see very less number of cells that are there after the treatment with the endophyte bioactive compounds. So this is one more activity that you can do. Next please. Then India is now the hub of non-communicable diseases. रोज शाम के हम निकल जाते हैं, कभी मोमोस खा रहे हैं, कभी पेटीज खा रहे हैं, कभी बर्गर खा रहे हैं, all leading to obesity. The best example is speaking to you. So the charity begins at home. We took this project of finding anti-obesity compound, और obesity के लिए जो best target होता है दैट इज लाइपेज, क्योंकि लाइपेज ही जो है वो रिलीज होके ट्राइग्लिसराइड्स में होगा। तो अगर हम उसको रोक देते हैं, उसकी एक्टिविटी को रोक देंगे, तो बॉडी में फैट बनना नहीं होगा। सो मोस्ट इंपॉर्टेंट टारगेट फॉर एंटी ओबेसिटी इज द पैंक्रियाटिक लाइपेज। आप उसको ब्रेक कर दीजिए, आपकी बॉडी में फैट बनना कम हो जाएगा और आप मोटे नहीं होंगे। ओनली अवेलेबल ड्रग फॉर एंटी ओबेसिटी दैट इज FDA approved is orlistat, जेनिटल ने बनाया हुआ है। उसके अलावा सारी ड्रग्स मार्केट से वापस ले ली गई है बिकॉज ऑफ वन और द अदर साइड इफेक्ट्स। ओके, सो विल सी हाउ। नेक्स्ट प्लीज। तो इसकी स्क्रीनिंग के लिए बस याद रखिए, आपके पास वो 1 mg पर ml वाला स्टॉक है, या तो आप डायरेक्टली उसको ले सकते हैं या ब्रॉथ ले लीजिए जिसमें कोई भी वो एनसीएम मेम्ब्रेन के थ्रू पास हो ताकि बैक्टीरियल कंटेमिनेशन ना है। So 2 ways you can do. One is the olive oil rhodamine B plate essay and one is the phenol red olive oil plate essay. Okay? So हमारा सबस्ट्रेट यहाँ पे है olive oil और दो इंडिकेटर्स है, एक रोडामीन है और एक फिनॉल रेड है। नेक्स्ट में आएंगे, हमने क्या किया, ऑलिव ऑयल को लिया, उसके साथ अपने एंडोफाइट्स जो है उसके बायोएक्टिव कंपाउंड्स को इंक्यूबेट किया, उसके अगेंस्ट एक रिस्पेक्टिव कंट्रोल रखा जिसमें कल्चर फिल्ट्रेट नहीं था, सिर्फ वाटर था या ब्रॉथ था, और उसके साथ हमने क्या किया, ऑलिव ऑयल को रखा। ऑलिव ऑयल को रखने के बाद हमने उसको इंक्यूबेट कर दिया फॉर 37 डिग्री फॉर 24 आवर्स, और उसके बाद हमने क्या किया, रोडामीन प्लेट में हमने यूवी रेंज मार के देखा, तो क्या हुआ, यू विल सी द हेलोस। ओके, तो जहां भी लाइपेज एक्टिविटी होगी, मतलब कि जहां पे इनहिबिशन नहीं हुआ है, we can see there is zone formation, है ना। और जहां पे भी एंटी ओबेसिटी एक्टिविटी होगी तो क्या करेगा, वो लाइपेज को ट्राइग्लिसराइड में होने से रोकेगा। सो यू विल सी कि यहाँ पे जोन नहीं बन रहा है, यहाँ पे जोन नहीं बन रहा है। जहां पे जोन बनना कम हो गया, देयर यू कैन से नाउ दिस इज माय एक्सट्रैक्ट और एंडोफिटिक कल्चर ऑफ इंटरेस्ट। नेक्स्ट प्लीज। यहां पे भी देखिए, जहां पे ये फिनॉल रेड है, जैसे ही पीएच चेंज होगा, फैट्स बनेंगे तो पीएच चेंज होगा, जैसे ही पीएच चेंज होगा क्या होगा, जोन साइज बनना चालू हो जाएंगे, जोन दिखने चालू हो जाएंगे आपको येलो कलर के। बट जहां पे एक्टिविटी है, you can see here there is no zone formation. Why? Because they are not changing into the triglyceride. So based on that, next आइए, you can say that they are inhibiting the pancreatic lipase, and we found two very important and very potent endophytic fungi. One is H6 AM and WLS. AM stands for Agel marmalose, L for leaves, and WLS stands for Wynath, life century. Similarly, 57 TBP ALM stands for Texas baccata, bark almoda. Okay, so इन दो compounds को हमने लिया, दो endophytes को लिया. Next please. And we did the quantitative screening using para-nitrophenol laurate, जो कि रिएक्ट करता है सेम तरीके से विद एंडोफाइट एंड रिलीजेस पैरानाइट्रोफिनॉल। उसके बेसिस पे व्हाट वी हैव फाउंड दैट सिमिलर और बेटर टू एंडोफाइट, orlistat is the anti-obesity drug which is giving IC50 at 2.73 mg per ml, such low concentration, and our endophyte is giving इससे भी low concentration. So we found something which is very potent, and we moved on with this compound. So this is another aspect of endophytic fungi which you can tap and go ahead. Next please. Okay, since we are talking about non-communicable diseases, obesity is a gateway to many diseases. कैंसर भी होता है, कैंसर के साथ बीपी की प्रॉब्लम भी आती है, बीपी के प्रॉब्लम के साथ हार्ट डिजीज भी आती है। ओके, हार्ट डिजीजेस में आपको अगर क्लीनिंग करनी है, जैसे अगर हमारा वॉश बेसिन में कुछ फंस गया है तो हम उसमें विनेगर वगैरह ऐड करते हैं या दूसरे सॉल्ट्स ऐड करते हैं तो क्लीन अप। ओके, वैसे ही हम क्या करेंगे, अपने एंडोफिटिक फंजाई को लेंगे और उसके एक्सट्रैक्ट को यूज करेंगे फॉर क्लीनिंग अप द क्लॉट्स दैट आर फाउंड। और क्लॉट्स क्यों बनते हैं, बिकॉज ऑफ द क्लॉटिंग कास्केड, जिसमें से फिब्रिन के चंक्स बन जाते हैं इन द वेसल्स। और जैसे ही किसी को हार्ट अटैक आता है, सबसे पहले थ्रोम्बोलिटिक ड्रग दी जाती है ताकि वो क्लॉट जो है वो हट जाए और ब्लड फ्लो शुरू हो जाए। ओके, So anyone knows what is the contribution of Indian science in formation of thrombolytic drugs? आप लोगों ने सुना होगा ना, स्टेंट बहुत महंगे होते हैं, सारा कुछ, एक डोज बहुत महंगी होती है. There was one scientist, late Professor Girish Sahani, who was ex-director CSIR and also advisor to our Honorable Prime Minister Shri Narendra Modi. He took out this challenge. एक डोज पहले कॉस्ट करती थी अराउंड 20,000 पर शॉट. अगर आपको एक लेना है तो 20,000 रुपीज स्पेंड करना पड़ेगा. सोचिए एक ऐसा देश जहां लोगों को हमको खाना देना पड़ रहा है पिछले 3 सालों से, तो सर्वाइव को 20,000 नहीं दे सकता. सो ही टुक अप दैट चैलेंज एंड उन्होंने वर्क किया उस थ्रोम्बोलिटिक ड्रग्स पे एंड ही फाउंड समथिंग न्यू जिसको उन्होंने नाम दिया स्ट्रेप्टोकाइनेस, डेवलप्ड बाय CSIR Imtech. It only cost 750 रुपए. But it took him 20 years to do that. सोचिए 20,000 रुपए से 750 रुपए में आपको एक ड्रग मिल रही है कैडिला की. सो दिस इज व्हाट. तो उन्हीं से इंस्पिरेशन ली हमने और कोलैबोरेट भी किया एंड वी फाउंड. नेक्स्ट प्लीज. तो दिस इज अ प्लेट वी फाउंड बाय मिक्सिंग फिब्रिन एंड थ्रोम्बिन. फिब्रिनोजन और थ्रोम्बिन को मिक्स करेंगे तो क्या होगा, फिब्रिन का आर्टिफिशियल क्लॉट बन जाएगा. हमने डिफ्यूजन ऐसे यूज किया. एंडोफाइट का जो कल्चर एक्सट्रैक्ट है उसको वेल्स में डाला, वी पुट इट अप देयर एंड देन वी अलाउड इट टू डिफ्यूज फॉर 24 hours. नेक्स्ट डे वी गॉट सर्टेन ब्यूटीफुल क्लॉट डिसॉल्विंग एक्टिविटी. Then we moved for characterization. So उसका क्या कहते हैं, SDS किया, native किया, and we found that there is one protein band monomeric somewhere around 37 kilodalton which was showing activity. तो ये जेल है, जेल mixed with fibrinogen and thrombin. And then हमने उसके ऊपर क्या किया, अपना एंडोफाइट का जो प्रोटीन था उसको रन किया. तो जहां पे एक्टिव साइट थी यू कैन सी कि वहां पे फिब्रोनोजन का क्लॉटिंग हो गई एंड यू कैन सी द एक्टिविटी. नेक्स्ट प्लीज. फिब्रिनोजन is made up of 3 units, alpha, beta and gamma. Okay, तो हमने टाइम इंटरवल में स्टडीज देखी. टाइम इंटरवल में स्टडीज देखी तो इनिशियल 5 मिनट्स में क्या होता है, अल्फा बैंड डिग्रेड हो जाता है हमारे एंडोफाइट से. 10 मिनट के बाद बीटा बैंड हो जाता है और गामा बैंड स्टेज इन कांटेक्ट टिल 3 hours. सेम विद द प्लाज्मिड जो स्टैंडर्ड है. So by looking at this we can say it has alpha beta fibrinogenase activity. Next please. हमने उसका molecular weight लिया using Malditoff and we found it at 37 kilodalton which was also supporting our SDS page and other activity. Next please. हमने उसको measure किया using HPLC, so it was showing the similar kind of RT time with the plasmid. Next please. Next. We characterized it for what kind of activity it is having. So हमने inhibitors डाले different pH and different temperatures पे देखा and we found that it's a metalloprotease showing less and very good activity in comparison, sorry, more and very good activity in comparison to plasmid. आप KMVMAX देखिए तो कम सबस्ट्रेट पे ही उसका काम चल जा रहा है. ओके, फिर हमने क्या किया, हमने आर्टिफिशियल ब्लड क्लॉट्स बनाए. This is controlled without any treatment and this is gradual increase in the concentration. So at 50 microgram you can see the whole blood is dissolved. उसमें कोई भी clotting available नहीं है. This suggests that it is a very potent anti-fibrinolytic or fibrinolytic enzyme. Next please. Next. Okay, then we go for the taxonomy. So हमने ITS taxonomy की, ITS के अलावा LSU भी किया था, RSU भी किया था and we found that it's a Xyleria curta species. उसके बाद हमने उसकी N-terminal sequencing कराई which we found that it's a novel protein not reported till date and it's a peptidase and a protease enzyme. Okay, और उसके बाद हमने उसका normal cells में टेस्ट किया, raw murine macrophage टेस्ट में 267 cell lines पे and we found that at 50 μg concentration it is not having any toxic activity against normal cells, so it can be further taken for क्लिनिकल और सब क्लिनिकल ट्रायल्स। नेक्स्ट प्लीज। ओके, अभी तक हमने बात की सारे फार्माकोकाइनेटिक्स के बारे में। अब बात करते हैं थोड़ा डिफरेंट एस्पेक्ट के बारे में। जस्ट गिव मी अ मिनट। जितना भी खाना हम प्रोड्यूस करते हैं या जितना भी एग्रीकल्चर प्रोड्यूस होते हैं, आपको लगता है इनफ है या हम अभी भी लैग कर रहे हैं? वॉट एवर वी प्रोड्यूस, उसका 40% लॉस हो जाता है पोस्ट हार्वेस्ट में। मतलब फील्ड में प्रोड्यूस हुआ और आपके खाने के प्लेट में आते तक 40% लॉस हो जाता है। क्यों? क्योंकि उसमें डिफरेंट टाइप के इन्फेक्शन हो जाते हैं और उस इन्फेक्शन को बचाने के लिए फ्यूमिगेंट्स का यूज किया जाता है। बट फ्यूमिगेंट्स आर कार्सिनोजेनिक एंड वेरी टॉक्सिक टू नेचर। तो हमने क्या किया? वी टुक अप दिस चैलेंज and we isolated this novel 6-7 muscular species from India, from northeast and southern part of the India. Next please. And following in the footsteps of Subramaniam Sahab, हमने अपने मस्कुलर का नाम रखा कषायम। कषायम मीन्स पंजेंट, वेरी तीखा। ओके, वो भी संस्कृत में ही नाम रखते थे। तो उसका हमने एनालिसिस किया। नेक्स्ट प्लीज। नेक्स्ट। Yes. So you can see, if there is no muscular species, the fungi is, this is the plant pathogen, जो कि food spoilage cause करता है. It is growing very well. But when there is muscular species, it is producing certain volatiles which is inhibiting the growth of the fungi even after 7 days of incubation. Next please. उसको फिर हमने fruits में testing की. So you can see these are grapes inoculated with what varieties? No fungi. You can see the spoilage happening here. But when there is fungi that is producing volatiles, you can see all the plates are in very good, very intact condition. Same with the strawberries. When the fungi is not there, it is producing, it is leaving the strawberries to rotting. But when the fungi is there, it is letting the fruit to stay in a very good condition. अच्छी बात ये है that it is completely sterile. उसको कितना भी आप न्यूट्रिशन डेफिशिएंट मीडियम में डाल दीजिए, यूवी दे दीजिए, कुछ भी कर दीजिए, वो स्पोर्स नहीं बनाता। स्पोर्स नहीं बनाता तो फैलने के चांसेस नहीं है। तो इसके लिए माय पीएचडी सुपरवाइजर हैज सेट अप अ स्टार्टअप एंड वी आर नाउ almost about finishing its commercial production. We are in almost the last stage of commercial production. So maybe next few days आपको आ जाएगा। बहुत सिंपल है। गेहूं लीजिए, राई लीजिए, उसमें एंडोफाइट डाल दीजिए और आपके पास जो टी बैग्स होते हैं ना, उसके अंदर रख दीजिए। 20 पैसा कॉस्ट आती है, 20 पैसा, उसको रख दीजिए और जब आपको फ्रूट्स रखने उसमें हल्का सा पानी डाल दीजिए टी बैग में। जैसे ही पानी डालेंगे क्या होगा? इट स्टार्ट्स एमिटिंग वोलेटाइल्स। तो क्या होगा? एक एनवायरनमेंट पूरा बन जाता है जिसमें फंगस ग्रो नहीं हो पाती। दिस इज व्हाट इट इज टू मी। ओके, नेक्स्ट। अपार्ट फ्रॉम दैट, दिस इज व्हाट आई हैव डन फॉर माय पोस्ट डॉक्टरल स्टडीज। तो एग्रीकल्चर में कैसे हो सकता है? वन ऑफ द मेजर प्रॉब्लम द वर्ल्ड इज फेसिंग इज सैलिनिटी। ऑलमोस्ट 50% ऑफ द लैंड बाय 2050 विल बी सैलाइन, और उसको हम कैसे बचा सकते हैं? नेक्स्ट, नेक्स्ट, नेक्स्ट। ओके, सो दिस इज द मैप ऑफ इजराइल और हमने साउथ का जो एरिया है, इट इज सिमिलर टू व्हाट वी हैव इन लद्दाख। डेजर्ट है, कोल्ड डेजर्ट है, दिन में बहुत गर्मी होती है। लद्दाख में तो नहीं होती, बट वहां दिन में बहुत गर्मी होती है और रात के ठंड होती है। ओके, सो व्हाट वी डिड, हमने वहां जो वाइल्ड ग्रोइंग प्लांट्स है उनको लिया, विच आर नॉट, और हमने मेलन्स के अगेंस्ट टेस्ट किया था, जो तरबूज और खरबूज खाते हैं ना, उसके अगेंस्ट। तो जो वहां वाइल्ड ग्रोइंग प्लांट्स हैं, दोज आर नोन एज स्क्विटरिंग मेलन्स और स्क्विटरिंग कुकम्बर्स, उसको लिया। क्यों? क्योंकि अकॉर्डिंग टू द हाइपोथेसिस, याद करिए क्लास 2 लेवल में क्या था कि हैबिटेट एडॉप्टेड है। अगर जिस हैबिटेट में है तो वहां के लिए अडॉप्ट हो जाएगा वो एंडोफाइट और इट विल गिव द एक्टिविटी। हमने वहां से आइसोलेट किया। नेक्स्ट आइए, और वी गॉट दिस ब्यूटीफुल फंजाई। नेक्स्ट, 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 नेक्स्ट होते जाइए ना, पीछे आइए, पीछे आइए, पीछे आइए, पीछे और पीछे। ओके, सो हमने स्क्रीनिंग चालू की. This is PDA plate without any salt. This is PDA plate with 100 millimolar, 200 millimolar और 500 millimolar. होना क्या चाहिए था, जैसे जैसे सॉल्ट कंसंट्रेशन बढ़ रही है फंजाई की ग्रोथ कम होनी चाहिए, बट ये जीवट है, उल्टा है, जितना सॉल्ट दे रहे हैं उसमें ज्यादा एक्टिव होके ग्रो हो रहा है. फिर हमने उसको प्लांट में इंट्रोड्यूस किया. दीज आर कंट्रोल प्लांट्स विदाउट एनी एंडोफाइट ट्रीटमेंट. 100 millimolar, 200 millimolar. So what you can see, जहां एंडोफाइट नहीं है, the plant is growing after 3 weeks of salt treatment. इसको हमने नमक पानी दिया 100 millimolar and 200 millimolar. लेकिन जहां पे एंडोफाइट है, you can see how beautifully it is mitigating the salt stress. You can see here, यहाँ कोई भी क्लोरोसिस नहीं है, किसी भी टाइप का डिजीज सिम्टम्स नहीं है. Next please. So you can see how it is increasing the fresh root and the dry root mass. Also the leaf damage is controlled by 40 to 50%. Next. फिर हमने उसको फील्ड में ले गए. Next. And you can see कि in compare to the control, there is always a better activity in export and the import quality of the melons. और ये सारा melons जो है ठंड के टाइम में यूरोप में जो होता है इसको भेजा जाता है फॉर कमर्शियल पर्पस. नेक्स्ट अनदर. सो दिस इज ऑल व्हाट वी हैव डन. तो जहां जहां एंडोफाइट होता है, यू कैन सी जहां जहां एंडोफाइट है वहां फोटोसिंथेसिस अच्छा हो रहा है. एंडोफाइट नहीं है तो फोटोसिंथेसिस अच्छी नहीं हो रही है, एंड द प्लांट लिटरली डाइज. जहां एंडोफाइट है वहां पे सोडियम का कंसंट्रेशन कम है, कम ले रहा है सोडियम. पोटेशियम का बढ़ गया, बिकॉज पोटेशियम इज गुड फॉर growth of the plant. Also chloride concentration कम हो जाता है in comparison to the control plant. Next. ये देखिए, this is control. 100 millimolar, 200 millimolar Ross generation H2O2 डालेंगे तो देखिए कैसे स्पॉट्स आ रहे हैं. इसमें देखिए जब एंडोफाइट से थे स्पॉट्स कम हो गए. सेम विद द सुपर ऑक्साइड आयंस, also with the mortality. So you can see, and importantly हमने लिग्निन डिपोजिशन देखा. जैसे जैसे लिग्निन का डिपोजिशन बढ़ता है, स्ट्रक्चरल फ्रेमवर्क ऑफ द प्लांट इंप्रूव्स. तो जहां एंडोफाइट्स है, यू कैन सी हमने इसको डाई से स्टेन किया तो यू कैन सी कि लिग्निन कंसंट्रेशन इसके कंपैरिजन में ज्यादा है. नेक्स्ट. और उसके बाद हमने फ्लोरोसेंस माइक्रोस्कोपी की, एंड आफ्टर 21 डेज यू कैन क्लियरली सी कि एंडोफाइट्स जो है these red colors are endophytes colonizing. This is the root all. तो आप देखिए जैसे जैसे सॉल्ट कंसंट्रेशन बढ़ रहा है वैसे वैसे colonization बढ़ते जा रहा है. This means this endophyte prefers salt. Next please. Next is Fusarium oxysporum, one of the most devastating fungal pathogen जो कि wilting cause करता है, रूट्स को colonize करता है. जो भी न्यूट्रिएंट्स है वो ऊपर जा नहीं सकते और उसके कारण विल्टिंग हो जाती है. तो आप देखेंगे दिस इज फ्यूजेरियम स्पीशीज एंड दिस इज एंडोफाइट. सो यू कैन सी एंटीबायोसिस हैपनिंग हियर. ये कंट्रोल है, इस देखिए कैसे ग्रो हो रहा है. इन करस्पॉन्डेंस टू दैट यू कैन सी ए इनहिबिटरी आर्क फॉर्मिंग हियर. ओके, तो इसके बेसिस पे हम फिर आगे बढ़े. नेक्स्ट प्लीज. और आप देखेंगे आफ्टर 21 डेज दिस इज कंट्रोल प्लांट. फ्यूजेरियम ओनली विद प्लांट. This is control nothing. This is control plant with endophyte. This is plant with fusarium. This is plant with endophyte and fusarium. 21 दिन के बाद देखिए कि ये मर चुका है या जिंदा है. कुछ भी नहीं हो रहा है प्लांट को. It grew up to 45 days very well. और 40 से 45 दिन में इसकी पहली फ्रूटिंग हो जाती है. तो देखेंगे फिर डिजीज सीवियरिटी, डिजीज इंडेक्स सारा जो है वो कम हो जाता है. नेक्स्ट प्लीज. नेक्स्ट. All these characters we have identified and similar observations were there. Next. आ जाइए next. यहाँ भी सेम चीज है. नेक्स्ट आ जाइए. Same. This green one is Fusarium oxysporum and this red one is our endophyte. And you can clearly see जब हम उसका extract डालते हैं तो this is inhibiting the fungi in comparison to the control. ये control है, ये with fungal extract. So how the pathogen is increasing. Next आइए. फील्ड ट्रायल्स लगाए हमने. ऑब्जर्व. नेक्स्ट. सेम रिजल्ट्स है. वी फाउंड व्हाट वी हैव फाउंड फॉर द अदर वन. नेक्स्ट आइए. तो अदर एक्टिविटीज हमने चेक की, फॉस्फेटेज चेक की. नेक्स्ट होते हैं कंट्रोल में देखिए ब्लू कलर है. जब वो फॉस्फेटेज प्रोड्यूस करेगा तो अलग हो जाएगा, हार्मोन्स अगर प्रोड्यूस कर रहा है तो साल्टोस के डालने के बाद इट विल प्रोड्यूस पिंक कलर। नेक्स्ट सेल वॉल डिग्रेडिंग एंजाइम्स बनाता है व्हिच हेल्प इट टू कॉलोनाइज बेटर, सेलुलेज बना रहा है, प्रोटीएज बना रहा है, काइटिनेज बना रहा है। नेक्स्ट है ये फंगल पैथोजन को रोकने के लिए सबसे इंपॉर्टेंट होता है साइडोफोर प्रोडक्शन। यू कैन सी द एंडोफाइट इज आल्सो प्रोड्यूसिंग साइडोफोर्स। नेक्स्ट फिर हमने देखा कि व्हाट आर द कंपोनेंट्स ऑफ दिस एंडोफाइट, सो वी फाउंड दैट इट इज प्रोड्यूसिंग, नेक्स्ट आइए, different types of compound। next आइए, so this is the heat map of all the compounds that are there। next हो जाइए, next so पीछे आइए, so this is what it is actually producing, you can see। और हम लिटरेचर में अगर जाके देखें, so you can see benzene 1,3-bis, this is already reported to have antibacterial activity। all these compounds produced by endophytic fungal fraction, 11 fractions of what we have tested is producing this, methyl stearate 6, all these oxalic acid 5, so these are already having different kinds of activity that is reported in the literature। next, और जब हम देखते हैं कि होस्ट प्लांट में क्या बन रहा है और हमारे एंडोफाइट में क्या बन रहा है, तो यू कैन सी कि यहाँ यूरोसिलिक एसिड बन रहा है, यहाँ उसका आइसोमर बन रहा है, यहाँ पे पाइपरीन बन रहा है, यहाँ उसका आइसोमर बन रहा है। So this suggests that endophytic fungi not only produces non-analogous compound, it also produces analogous compounds that are produced by the host plants। next, so what we can conclude from this, these endophytes are actually the microfactories within the plant that are having various kinds of activities, and this should be taken into account। अभी हम बिल्कुल नए हैं, it is only 20 to 30 years old branch that you can take up the new plants, new medicinal plants, non-medicinal plants for different kinds of activity। next, these are the references। no research would be possible, and I'm very thankful for these people, my PhD supervisor Professor Sanjay Saxena, my Dr. Stanley Freeman, Supla Sahab, my guide, my mentor, Dr. Girish Sahani, who is no more with us, and to the father of endophytic fungi, Professor Gary Strobel, who has been instrumental in motivating and guiding us, and two gentlemen sitting over there, my gurus, जिनके पास सबसे पहला कॉल जाता है कि सर मैं यहाँ फंस गया, उसका सॉल्यूशन क्या होगा, हांडे साहब है, अमित सर, who has always been there to rescue me। Thank you, sir। And we are organizing a conference in our university in 26th and 27th, 28th of February, so you all are invited। It's in a hybrid mode, so please join us for the conference। Thank you।
Excellent।
Questions, please।
Questions?
Ma'am, comfortable। all these protocols, whatever is there, go to Google Scholar, all this is published, तो आपको मेरे सारे पेपर्स Google Scholar में मिल जाएंगे। If you want any kind of help from me, you are more than welcome to contact me। Okay, my email ID is with Khande sir, है ना? आप please, हाँ, questions please।
Sir, as you mentioned in your result, Alternaria, Fusarium, you isolated as an endophyte, but
Yes.
it also act as a pathogenic fungi।
Yes।
So what is the difference between this?
Yes। देखिए, जब भी पैथोजेनिक इंटरेक्शन होगा ना, आपको प्लांट में सिम्टम्स दिखेंगे। जब भी एंडोफाइटिक होगा, प्लांट हरा दिखेगा, बिल्कुल उसमें कोई भी इंफेक्शन नहीं होगी। आप क्या करते हैं जब आइसोलेशन लगाते हैं? दैट्स अ वेरी गुड क्वेश्चन। चलिए ना, वेरी बेसिक क्वेश्चन। जैसे ही आप सरफेस स्टरलाइजेशन कर लेते हैं, तो एंड में जो आपका सरफेस स्टरलाइज्ड पानी बचता है, या 30% इथेनॉल या 40% इथेनॉल जो भी यूज कर रहे हैं, उसका 100 माइक्रोलीटर लीजिए और उसको एक पीडीए प्लेट के ऊपर स्प्रेड कर दीजिए, स्प्रेडर से स्प्रेड करिए, इंक्यूबेट इट विद योर टेस्ट प्लेट्स। क्या होगा? अगर कोई चीज एपिफिटिक है तो वो कंट्रोल प्लेट में ग्रो हो जाएगी, है ना? जैसे ही एपिफिटिक होगी, अगर मान लीजिए वो अल्टरनेरिया एपिफिटिक था, तो क्या होगा कि वो उस कंट्रोल प्लेट में ग्रो हो जाएगा। सो यू रिड्यूस दैट एंड कलेक्ट द अदर फंजाई। आपकी जो बात है कि वो पैथोजेनिक हो सकता है, बिल्कुल हो सकता है, क्या होता है जब आप किसी से हेल्प मांग रहे हैं तो एक कोरिलेशन बनता है, ठीक है। यहाँ पे यूज होता है, बॉयल ने 2005 में एक टर्म दिया था बैलेंस्ड एंटागोनिज्म, उसको वो फॉलो करके क्या करता है, जब तक होस्ट प्लांट की इम्यूनिटी अच्छी है तब तक उसको कोई भी नेगेटिव इफेक्ट नहीं कॉज करता। जैसे ही होस्ट प्लांट क्या हो जाएगा कि वो बूढ़ा हो जाएगा या उसकी इम्यूनिटी डिक्लाइन हो जाएगी, देन वो अपने आप को सर्वाइव करने के लिए इट कैन स्टार्ट शोइंग पैथोजेनिक सिम्टम्स टू इट, और फिर वो उसको एज अ कार्बन सोर्स यूटिलाइज कर देता है। थैंक यू।
Thank you, Vinit sir. It was very nice journey to listen you on the topic fungal endophytes from nature's Ichacus to macrofactories of the bioactive compound. Really, it was particularly, I will say personally for me, it was great to listen this topic because nearly four students are working under my supervision on this topic of endophytes. And these students are working particularly the local plants from our Vidarbha region, that is, one is working on the Tridax procumbus, Solanum xanthocarbon, one is working on the Clitoria ternitia, Cassia tora, and another one is working on the Barbelia. So these are these plants we have taken from this Vidarbha region, and they are exploring, but very new things I have learned from this lecture, particularly like the fumigation technique, that is many things which I have noted, very new things. So I will start from your first part, that was according to the definition, means what exactly meaning of the endophytes. So that was elaborated, well elaborated. As you said that it was explored after the discovery of Taxol from Taxomyces, that was from the plant Gymnosperm plant Taxus. So it was explored by the Gary Strobel, and after that there was say huge uh, running towards this exploration of this discoveries or the experiments of these endophytes. Nowadays, very few workers are working on this particular topic, and this is the need, one can say. अभी जैसा आपने बताया कि यहाँ पे बहुत सारे ऐसे पैरामीटर्स है जिस पैरामीटर्स पे अपन वर्क कर सकते हैं. फॉर एग्जांपल एंटी कैंसरस ड्रग्स जो आपने बताया, एंडोफाइट्स एंटी, आपने जो नाम लिया जाए, फॉर एग्जांपल विंकास्ट्रीन. सो विंकास्ट्रीन इट इज जनरली एक्सट्रैक्टेड फ्रॉम द प्लांट विंकारोजिया, ओके, सदाफुली. वहां से उसको एक्सट्रैक्ट किया जाता है, लेकिन उसके अंदर में रहने वाली जो फंजाई है, जिसको अपन एंडोफाइट्स फंजाई बोलते हैं, उसको यूज करके अगर आपने विंकास्ट्रीन एक्सट्रैक्ट किया तो वो प्लांट जो है, वो प्लांट से ज्यादा उससे विंकास्ट्रीन निकल सकता है. नॉट ओनली दिस, टैक्सस जैसे जो प्लांट है, जो प्लांट अभी एक्सटिंक्ट होने के इस पे वर्ज पे है, एक्सटिंक्ट हो सकते हैं कि ओवर एक्सप्लॉयटेशन से, वो भी बच सकते हैं ड्यू टू द यूज ऑफ दिस endophytic fungi which are extracted from such type of novel plants. Then you have mentioned that there are uses of, particularly I will mention some uses like anti-cancerous activity, and one thing that is about the anti-obesity compound. ये तो नई बात सुनने को आई कि एंडोफाइट से एंटी ओबेसिटी कंपाउंड भी एक्सट्रैक्ट हो सकता है. सो वेरी नाइस. दूसरी बात जो आपने, और एक चीज आपने यहाँ पे ऐड की, दैट इज रिगार्डिंग दी नोवेल क्लॉट ब्लस्टर फ्रॉम द एंडोफिटिक फंजाई. जो सुनते जा रहे हैं हम, वी आर वेरी सरप्राइजिंग. बैठे हुए जितने भी लोग हैं, तमाम लोग हैं, ये सुन के, क्योंकि नई चीज आज पढ़ने को मिल रही है, सुनने को मिल रही है कि फंजाई में कितना पावर है कि आपके हार्ट को अच्छा कर सकता है, आपके ओबेसिटी को दुरुस्त कर सकता है, इवन आपका अगर कैंसर होने वाला है उसको रोक सकता है. तो दिस इज द पावर ऑफ द फंजाई, पर्टिकुलरली एंडोफिटिक फंजाई. और जितने भी यहाँ पे यंगस्टर्स बैठे हैं उनको तो मैं बोलूंगा अभी कि इसके ऊपर ज्यादा वर्क करो. यहाँ पे मेरे स्टूडेंट भी बैठे हैं जो अभी, जिनको मैंने एमएससी के लिए भी टॉपिक दिया वो प्रोजेक्ट का एंडोफिटिक फंजाई के ऊपर, तो उनको मैं ज्यादा एक्सप्लोर उसके ऊपर करने के लिए बोलूंगा अभी. और जितने भी पीएचडी के यहाँ पे स्टूडेंट्स होंगे वो अभी उसको ज्यादा एक्सप्लोर करो, जिससे जैसे सर ने बताया कि नोवेल क्लॉट ब्लास्टर और दूसरा जो है सैलिनिटी का प्रॉब्लम, सर ने तो एक बहुत बड़ा आता है, क्योंकि हमारे इधर में अमरावती साइड में देर इज वन पर्टिकुलर रीजन जिसको खारपान पट्टा बोला जाता है। तो उस खारपान पट्टे में हमारा अमरावती का कुछ पार्ट आता है, अकोला डिस्ट्रिक्ट का कुछ पार्ट आता है, तो उसमें खेती जो है वो ज्यादा ये नहीं होती, यील्ड नहीं होता। उस पर्टिकुलर उसमें रीजन में ऐसे अगर कुछ किया जाए, एक्सपेरिमेंट्स किया जाए, सो दैट विल बी वेरी यूजफुल फॉर द फार्मर्स। सो यहाँ पे जो जितने भी बैठे हैं, इवन नागपुर से भी कोई अगर इंटरेस्टेड है स्टूडेंट, तो वो भी कर सकता है इसके ऊपर काम। और सर खुद बोले कि वो अपने को ट्रेनिंग भी दे सकते हैं। तो ये बहुत ही मोटिवेटिंग आपने जो बताया, इट वॉज वेरी मोटिवेटिंग स्पीच एंड द लेक्चर विच वॉज डिलीवर्ड बाय डॉक्टर विनीत मेश्राम सर। On behalf of MSI Amravati unit and on behalf of this college, as the chairman of this session, I am very much thankful to you for delivering such a nice lecture. Thank you.
Thank you very much, sir. A very big thank you to Dr. Meshram sir for delivering a very nice and informative lecture in technical session 2. Now onwards there is a lunch break. So I request all the honorable dignitaries and the delegates and participants of this national seminar that lunch is arranged in the our college hostel mess. So all are requested to visit our college hostel mess for the lunch. After lunch, there is the hands-on training session in botany department's laboratory. So all the participants and delegates are requested to visit botany laboratory for hands-on training session after the lunch. And after hands-on training session, there is a poster presentation in block B wing.